So I can see them here. Yes, sir. There is.
Good. So I'm going to give just a few slides as introduction of our center. And we had a very exciting and productive day yesterday, actually started on Sunday with very intense scientific discussions and collaborations on a variety of projects that our center is charged to pursue. And I think what I would describe yesterday from a student who said to me, there's so much excitement in the air in this room that I'm having all my heartbeat up at the whole time. So this is actually a very good representation of what's going on with the projects organized in our center. You could appreciate the title of our proposal is a community effort to translate protein data to knowledge. We are creating an integrated platform. We have had a dream. The dream is to end the fragmented information era of protein data science. A dream to create an integrated platform that would contain a variety of tools to serve both the proteomic community as well as all other biomedical communities at large. And we are so fortunate that NIH answered, and we are given this opportunity to actually realize the dreams and the goals that we want to pursue. My sincere appreciations is to the entire investigator team. Your talents and your contributions and dedications made this proposal submission a success. We owe it to you to make the execution of all the studies we propose a success. This is an opportunity for me to also introduce some of the key investigators in this center, starting from Henning Jacob. He's a cluster leader at EMBO EBI, sitting in the audience. And then Dr. Andrew Su, who's sitting right next to Dr. Carol Watson, and he really has been quoted as the prince of crowdsourcing and text mining in biomedical science. And Carol, right there, who you've met, as well as Dr. Mary Lindsay, sitting next to Dr. Wei Wang. Mary and myself have been together working in the field of cardiovascular proteomics, and she and I have been collaborating on a variety of projects in the past 10 years. So Mary's the one brought in one of the key clinical cohorts, Jackson Heart Studies, a population of 5,017 African American with cardiovascular diseases. And next to that is Professor John Yates. John and I have worked together since in the late 90s. I owe everything I know about proteomics to John, and his guidance and the mentorship and the friendship have been unreal for the past 15 years we've worked together. And I can keep going on with all the pictures, and some of them you know. And I do want to make a mention of, in particularly, our partner, Dr. Alex Booty, who was going to be here, but he's out of town today. And he has sent me his congratulations. And Alex and ourselves have been working together on a variety of organization of BD2K activities at UCLA campus. I will introduce all our partners in the engineering school. Dr. Carlos Zanino sitting over there, and Dr. Wei Wang, and Professor Jason Kang and I have known each other for more than 30 years. So their contributions made it critical for we have a solid computer engineering group participation in this Big Center Award. We do have our partners from Sage Bionetworks, from Seattle, Michael Cullen representing, also Stephen Friend, and we have a number of our also collaborators from the San Diego area that participating today. You've seen the slides where we put forward to introduce the overall research plan. It is a center. The research plan are both comprehensive as well as grand. And there are overall 9 scientific specific aims in the DSR section, and there are 3 parallel training modules in the education and training cluster. On top of that is an integrated consortium activities under the leadership and guidance of NIH BD2K initiative. If I could actually summarize all the tasks and milestones into 2 actions, the first actions would be building tools. Our center is charged to build a number of tools, ranging from tools that could aggregate, enhance, and further elevate the utilities of existing proteomic resources, metabolomic resources, and a number of other text mining tools that we currently have, as featured in one of our slides: GeneWiki project, Reactome project, BioGPS project, KabaKD project, Proton projects, all of which have been around for a number of years, but this is time by creating customized APIs. Those tools will talk to each other. There will be easy access for everyone in the community who need to have information annotated and accessed. We're also charged to build new tools. The center proposed to build up to 31 new tools in our software studio. They're ranging from analyticals of omics analysis, integration of text mining, phenotype analysis, integration of text mining both in PubMed and EHR. A number of tools will be built with the guiding principle of user-centric, community-driven, and serving our investigator community at large and globally. With all of these things proposed, we are at a stage where collaboration is the key. Collaborations and integrations being the second and equally important action item in our center, integrating not only just tools, but scientific approaches at all ESR training education levels is the second most important action item in the center. So with that, I want to tell you things have already got started. The excitement in the air didn't come from nowhere. They came from multiple well-organized center presentations that starts as early as 7:30 a.m. in California time. And our colleagues from UMBO EBI delve in on Fridays late afternoon to join these center presentations. Each of these presentations has been participated by 20 or 30 investigators in the center. And the first one started already earlier January this year. And so far we have had 8 of them. And these presentations and communications are going to continue throughout the year. We invite everyone who's interested to visit us, visit the center website. You could listen to all the tools that's been presented, as well as using them, putting in your feedback, and we will not only reply to your recommendations, we will customize changes while we go moving along the way. So I'm not going to repeat the research plan or the training plan as you've seen throughout the morning. I do want to say thanks to the Admin Center, BD2K website for the UCLA Center is up and running, and we appreciate the collaboration not only with CPSI, but also with QCB, led by Dr. Alex Hoffman, as well as the bioinformatics IDP by Dr. Yi Xin, in collaborating the training and education and research agenda of our center. With that, I think I hand it back to Anders.
Thank you, Dr. King. Well, as you can appreciate from the discussions that we've had in the last hour, it is the collective commitment of dedicated investigators in this room that truly defines the success and spirit of innovation in science. So I think that that concludes our introductory session here. I think that we're going to step out and go up to the courtyard if you'd like to grab a breath of fresh air, get some coffee and some snacks, and we'll meet back here in about 15 minutes. No?
I'm sorry. So as everybody can see on the agenda, the coffee break will actually be after our first plenary speech.
My mistake.
I didn't want to, give you guys any false representations.
On that
Yeah, all the people who attended the opening ceremony, thank you so much for taking the time out here through your busy schedules to share with us your wisdom. Thank you.
What's the title of this?
So and we can go up in the top one, we can go down, and then they can highlight the one that I'm writing there kind of right here. It really shows the core of what we're going to do. Now, this is not the lecture about mass spectrometry, of course, but I have to do some selection, and it's very exciting technology, of course. So, pioneers of this technology here in the room, John City, for example. Technology is really great in that it can do so many things these days. From proteogenomics, which I'll come back to the second half of my presentation, to more basic stuff such as differential expression, post-translation modifications on proteins, analyzing them on a large scale, of course, clinical applications such as surgical biomarkers or using a more basic way to understand the pathways regulated in disease. We work a lot on others, of course, and John mentioned that. Interactome uses technology to profile protein-protein interaction data. So all these are really exciting applications. All of these exciting applications require a lot of bioinformatics, and this is where my lab one contributes mostly. The technology itself, I have this slide because, again, it's not a, I guess, important to discuss the technology itself, but the technology is very nice, and it's beautiful and simplistic when you look at the slide like this. You have a protein, you have peptides, you generate electricity digestion, you ionize peptides, and then you have this technology that's under developed to measure mass to Charge ratio of molecules. So you can measure mass to Charge ratio of all fragments of a particular peptide, and then you can use computational tools to infer the peptide sequence for a particular peptide, and then protein then that generated that peptide. And it's amazing how many computational challenges technology like this can deal with, right? And that's what I've been working for, I don't know, years, and others work even longer. So again, a simple idea, a really, really exciting computation challenge. Just to give some history background to this field and where this help is coming from, how to identify peptides in technology by embedded searching. So this is actually, well, I think last year was the 20th anniversary of sequence, the first computational tool that was developed for enabling this type of analysis, or taking this type of mass spec data and identifying peptides with certain databases. So that's actually a contribution of engineering them yet. What we've been focusing on the last number of years is the provinces, like many technologies that best match to a spectrum, or any search that you do could be a true match that you're looking for, it could be a false match. And a lot of problems that we're dealing with in this field are related to the need to really separate the false stuff in the data from the true findings. And in this context, the best match to a particular spectrum that needs to require in the database could be a false peptide, a false indication of the wrong peptide. So it's quite a substantial problem. Again, kind of progressing in the history of the field that was a call to arms 10 years ago. These guidelines and these tools to deal with this challenge of analyzing this type of data and estimating the error rates that we have in mass spectrometry. And I contributed to establishing this data analysis guidelines that were adopted by most fields, most governments in this field. So given this background, I'll come back to that and I talk about the things that we're working on now that we're dealing with now. So, I mean, by now I think the challenges of the field of computational mass spectrometry are pretty well understood, and that range from the protein interest problem, similar to other fields such as RNA-seq. We have reads that map to multiple transcripts and have to deal with this. In the field of proteomics, we have peptides map to multiple proteins, so we have to deal with the shared peptides computationally. We have the error rates at different levels that we need to estimate. We have challenges that are more fundamental, such as the limited depth of analysis, in that it's hard to identify confidence of low balanced protein because we don't have very limited coverage of sequencing those proteins. We have other things that we and others are working on that are very relevant to what this center is like to achieve, such as, say, if you look for novel peptides or translational modifications, typically we're not looking for those in the research, and if we try to look for them, we have to be careful in how we do that. I will get back to that later in my presentation. So, well, if that's something that works, and now it's not. So something that is very important for this field and for all other fields is basically developing robust bioinformatics tools that can analyze this type of data. And so that if you have multi-study type of studies, such as well, pretty much most large-scale process non-long generation of data in multiple different places, and often the experiments are done using different technologies and different instruments, and data analysis using different tools. So the tools for basically computational tools for robust scoring of this data, regardless of how the data is generated, so that at the end, identification or classification of what's not will be compared across different studies and can be integrated in a meaningful way. I think is very important and a critical part. And we know that it's worked on that extensively for many years and developed certain strategies, some of them listed here, targeted or computational tools such as Rocket, for example. So I'm mentioning this just to talk in the next segment where I'll talk about the new challenges that we have now, these super large data sets and these integrative studies that we are dealing with. So I thought that for quite some time that a lot of challenges with the basic mass spectrometry and proteomics were actually solved. And that's when the field exploded as far as the sensitivity of the technologies and how much data you can generate. Now, whether you like this particular study or not, it's not the point here. The only point I want to make in this figure is that if you look at the number, 2001, this I guess from doing it, the point how many peptides you can identify per hour on a typical instrument, and there is an exponential curve where it's likely that you can get here with the new generation instruments. Again, my point here is that it's not maybe a revolutionary experiment sequencing as far as the speed of data generation, but it's certainly a pretty substantial progress that the field of proteomics is experiencing with the new instrumentation. And that brings us to this challenge of really the data sets are getting out of hand as far as the size of the data sets. Now, regardless of the improvement in the technology, we have large-scale data sets because we would like to do these experiments in replicates. We have multi-sample studies for biomarkers using label frequency where you just profile a lot of different experiments, a lot of different samples. We have large-scale interactive mapping efforts. And then we have this initiatives and efforts to actually pool all this publicly available data together as part of a data repository so that you can mine these data sets to extract biological knowledge from these data sets that wouldn't be apparent or possible to extract from a particular site that generated initial data sets. So all of these scenarios basically lead us through data sets that are extremely large in size and require really new thinking about how we can analyze them consistently. We have to redo a lot of the initial work that we've done on developing the informatics tools for this data. So again, experiments like this, where you combine many heterogeneous experiments from different research groups generated using different technologies, is a way to create a repository of, say, proteome profiles across different tissues or cell types. Requires really development of new computational strategies for processing the data and estimating error rates. So you probably, well, I'm sure you've heard about these studies, and again, we have differences of opinion on the studies in the field and on the pitfalls and advantages of doing this. But anyways, you could certainly try to attempt to integrate a lot of different data and combine the patterns that you observe across multiple different tissues in this integrated repository. So again, I'll come back to that when I talk about how we should do these things instead of how it was actually published. So to set this up, again, the challenges that we're experiencing, working that we need to work on is kind of this concept of understanding how the field progresses with the amount of data as the amount of data that's being collected is increasing. We are really moving from a situation where we were really identifying maybe a few thousand proteins per cell line of tissue to a situation where we identify close to 10,000 proteins. And also, when we integrate a lot of these different data types, we are really dealing with this over-sequencing problem. And again, I don't have too much time to explain that. I'll try to give you a hint on the next slide of what the computational challenge that we have here and why there is so much debate now about the error rates in this nature papers and other studies that are easy to publish. So the problem here is that as we go to this over-sequencing regime, where we have a lot of data that we collected, and we basically identifying pretty much every protein that could possibly be identified as a technology. But at the same time, the error rate, basically, the things that we can also identify in the protein databases that we're searching, they also keep accumulating. So we get into this unusual scenario where pretty much every sequence in the database that we search is identified by high-scoring hits, high-scoring peptides. And how we deal with this scenario of sorting out what's true and what's false in this regime is very different from where we were prior to certainly 10 years ago. Without trying to complicate things too much, we have to really think about this integrated multi-level modeling of performance data, where if we compute the protein score here at the end, based on all this evidence that we collect in a particular experiment, we have to come up with really a good understanding of what it means to actually identify a protein using mass spectrometry. And that we're not really identifying peptides, we're actually measuring mass to charge ratios for different ions for a particular protein, which have redundant information coming from different charge states of a particular peptide. So all of this kind of multi-level structure that gives information based on some evidence for a particular protein needs to be integrated to really say that based on all this data that collect a particular protein was identified its confidence, but how to do it really in the context of this super large scale data set is an open question now. Interestingly, for a long time personally in my research, we were thinking that one particular approach that works the best, and this is probably a bit to an extent for those who actually work with this type of data. It's a bit complicated concept, but basically we have a protein and we have many peptides that we identify for a particular protein in the experiment. How do we calculate the confidence in this protein based on all this peptide evidence, right? So you can come up with all these different computational approaches how you can do that. So we were advocating for one approach, combining the peptide evidence statistically from to calculate the protein score. And now when we revisit this, we look at this super large scale data set, and it seems like everything that we were thinking was the optimal approach before it to be revised, because many of the assumptions in the models of the disease are no longer valid. And we actually seem to be getting the best way to filter this type of data sets. If instead of combining the evidence from all peptides approaching the protein, we simply take the best peptide and just peptide and score for filtering. So that's kind of was something that was interesting for us, kind of almost going back to where the field was 10 years ago as far as the filtering of data was done. So anyways, what I'm trying to say here is that we need to rethink, and this is a slide that shows that that's the case using one particular data set. That we have to rethink a lot of things. Another challenge that we're dealing with now in the context of super large data sets, such as human protein project data sets, or those integrative or those composite nature papers, or methodological repositories, is the basic assumptions in the estimation of discovery rates. So there are tools such as BD2K strategies that people have been using for many years, and they need to be revised because and the assumption are no longer valid. And we have a proposal for policy and basic targeted strategies for FDR estimation have to be revised in case of very large data sets. So finally, I just want to make a few words about the proteogenomics. The field that we are really excited about, again, have to do it right, which means this is going beyond data collection to really extract the knowledge from these data sets. And one example is, of course, this use of proteomic technology to annotate, re-annotate genomic DNA sequences. So this is where, again, I wrote a review on this recently issued methods, where proteomics can be used as a great tool to basically look for novel things in the data. Peptides that can be predicted but have no personal evidence at this point. And that's what the concept of proteogenomics is. There is a lot of interest in this type of research, of course, because of the, sequence variants that are extremely important in various cancers, or because of non-coding RNAs and or RNAs thought to be non-coding or short-coding frames that claim to be very prevalent in many biological samples. So we are developing pipelines where we're doing this type of analysis where we build customized protein sequence databases from RNA-seq and genomics data, and then we search master parameter data against those databases, and then we annotate peptides according to what type of new knowledge they give us, whether it's a peptide supporting pseudogenes or re-annotating the gene boundaries for non-protein genes or supporting non-coding RNAs and so on. You can divide this type of peptides into different categories, as illustrated here, depending on what they map and how they map to a particular gene model. But again, coming back to the computational challenges of the BD2K initiative, this is what we need to think about and work on, is that, again, standard methods that we developed in the past for estimating the error rates and scoring this type of data, they're not applicable to proteogenomic studies. And if I had time, I would argue this slide in hand why that's the case. But I would advocate this class-specific focus error estimates that I'm trying to illustrate here, basically a different variant on the regular FDR that one has to do absolutely to get appropriately estimated error rates for novel peptides. And I illustrate here on this slide, taking this Nature Publications manuscript, and the peptides are reported there, and you can clearly identify certain issues in the data because the analysis that was done in those manuscripts was not taking into account challenges that I described here. But the good thing is that we are working on ways to do this right, and all this available information about variants and novel peptides isn't there. It's just we have to extract it with confidence, and I'm working on the guidelines, revising the guidelines where I started my presentation for scoring and analyzing protein data specifically for proteogenomics in this given part of the Nature methods manuscript for how we can analyze this type of data if we want to be confident about novel peptides and supporting implication of, say, similar questionable proteins. And this is just an example of how we're working with this. It's very complicated, but I just want to say that we're, bioinformatics is complicated, so we have to have these complicated workflows if you want to do things right. So this is just one of our complicated workflows that try to achieve this goal. So I think I'm running out of time, so I was going to make a few comments about some other things that we're working on, such as we're very excited about data independent acquisition technology, not a new concept. Again, John Gates contributed to establishing this concept quite a while back, but now it's used in a different light, I would say. And we develop, I think that there is a lot of promise in technology, and so we have some tools that we developed and also very recently published in Nature Methods. If anyone here is learning more about how to do that, I will be happy to talk to you later about this. So we think there are possibilities for bioinformatics innovative work and new ways to generate data in the field of mass spec as well. It's not just conventional shortcuts. So I stop here and acknowledge my lab, and I think a few minutes over time, so I don't know if you have any questions, but I'll be here and happy to discuss them.
Thank you for giving your talk. We do actually have about 5 minutes left for questions. If anybody's...
So one of the artifacts that we're dealing with now is looking at data across multiple levels. And so we have a lot of proteomic results, but then you alluded to in one of your slides the need to also put it in context of where that protein is in the cell, but then also where that cell is in the organ, et cetera. Can you tell us some insights that you have on how to cross scales with all of this data?
Right. So actually, I have several collaborative efforts that are trying to link, for example, cellular localization through a new technology called proximal interactome profiling, where they use this bio-idea approach and proximal biosimulation, where they can tag a protein and see what proteins come in proximity to the protein. And that's one source of data. It's pretty terrific that allows you to see protein interaction in a little bit different context, right? There are other localization studies that are now large scale data sets available. Of course, you can, with the cost of RNA sequencing, you can always do RNA sequencing as a part of your proteomics study, and that gives you a lot of information as far as, again, putting proteomics data, I guess, in the context of the transcriptome, gauge the ribosomal profiling data, and what this adds. So I think you're right that pretty much every study is going to be the type of study that will integrate in all this sort of information. How exactly to do it, I think this is where of course it depends on why we want to collect all this data. It's probably more, I guess, depends on the biological question at hand.
Has anybody done a comparison of when you use the RNA data to do searches versus what's the regular database?
We've done the comparison ourselves, and others are doing the comparison as well. So as far as the identification of peptides, you can use RNA sequencing as a way to constrain the database. Without even looking for novel peptides, novel splice forms, it's just simply asking the question, can we take a protein database with, I don't know, 40, 50, or 80,000 entries like 24, and constrain only those sequences for which they have RNA elements, and that you can basically match RNA elements. So that gives you not a whole lot, because it's basically just reducing the size of the database, and you benefit from improved sensitivity of peptide identification by basically searching against a small database. I wouldn't say that that's a huge advantage. With respect to novel peptides, you can see in RNA-seq data, yes, you can identify some, but I think the number of novel peptides, you certainly get less of 1%, which you would add to a normal universal person preference search. Less than 1% of extra things that you'll find would be novel peptides nominated by RNA-seq data. So my sense is that you're not going to get a lot as far as the numbers are concerned, but on the other hand, if you do get something that may be really important. So I think it's not the numbers, but looking at the importance of each thing that's going to matter.
For example, in the Liebler tumor study, was that approach able to find mutated sites?
So in there, they didn't use RNA-seq so much. I mean, they searched against the database that was a compilation of RNA-seq data, but it's a TCGA data, so it had a lot of information on what those variants, what variants they expect. So they already used, I think, a compilation of different sources, including known variants extracted from variant databases, protein databases, as well as genomic databases. I don't know how much of data identification really relies on just RNA-seq data that they also had. But I think that's an example of TCGA studies. If you do your own study, you would not have all this relative genomic information available for the same samples. So generating RNA-seq data certainly is something that would be pretty easy to do problem is, so again, so even a simple organic data set is going to nominate thousands, if not millions, of new things. Proteomics is only going to find a small fraction of that. But I think we shouldn't be afraid of the fact that proteomics will give you evidence for a very small number of things that RNA-seq nominates. It basically tells us that a lot of the stuff that you see in RNA-seq is not functional, it's not significant. So proteomics is a great tool to look at the functional significance of those things, even if the number is small.
Any more questions?
So I wonder if you can expand on the discussion of using specific FDR for local genomics. So is that because there are too many possible peptides, so you have to, treat that as separate?
Yeah, so this is quite specific FDR. It's very important concept to understand. And that if you have a novel peptide, the evidence that you need, that you have to require to call this accurately identified, the evidence should be much stronger than for a known peptide. So you cannot apply the same cutoffs, the same approaches to something that is likely to be in the data versus something that's very unlikely to be in the data. So that's a very basic concept, but for some reason it hasn't been applied in the field of proteogenomics. And I think a lot of those false positives that you see in the published papers are simply due to this misunderstanding of this basic concept.
Thank you, Dr. Steve. So now we're going to enter into our first coffee break, or I guess it's our second coffee break of the day, including registration. So we do have fresh coffee, and there's still some snacks available. We are going to be heading up to the courtyard for a couple minutes just to get us out of this room with no windows. So we'll convene back at about 10 a.m.
opportunities that we have here, right, is to identify some grand challenges that incorporate all the expertise in the room, and also advance sort of the center's mission in terms of big data. And so I want to describe what we, some of the thought process behind what we call the network of biotech. And to start initially, I just wanted to start with this analysis of this figure that I think really motivates the idea behind what we're doing. This just shows the number of PubMed indexed articles that are published each year. Currently, PubMed has over 24 million articles indexed. Over 1 million new articles are added every year. That's more than one new article every 30 seconds. So clearly, there's a huge expansion in the growth of the biomedical literature, and really underlying that is a growth in biomedical knowledge. But just because of the vast numbers, right, we as individual scientists can't consume that literature effectively. And in fact, right, PubMed is very hard to query and compute on, right? So if you have, for example, a query that is, I want to know all tyrosine kinase inhibitors that are relevant to hematologic cancers, right? And you do that search in PubMed, you get 496 articles that come back, which of course is a vast undersampling of what actually would be relevant to this particular topic. To really get all the articles you want, right, you need to enumerate all of the tyrosine kinase inhibitors that you are interested in. And you also need to enumerate all the hematologic cancers and then do the AND query, right? That is what really would come out. And it goes back to this idea that PubMed, while an incredible resource, we don't have efficient ways to access the knowledge that is contained within that resource. So you might argue that PubMed is an untapped source of big data. So the network of biothings is one attempt to try to make biomedical knowledge more computable, more queryable. The goal, simply stated, is to assemble a network of biomedical entities, where some of the biomedical entity types you might imagine are here: pathways, diseases, proteins, drugs, and so on. So assemble this network that is comprehensive, current, and computable. So how might we tackle the assembly of this network of biothings? Well, our strategy has always been, let's bring many disciplines to bear, throw the whole kitchen sink at it, from biocuration, text mining, crowdsourcing, citizen science, so on and so forth. These are all relevant to this idea of making knowledge more computable and accessible. Since there apparently is a kingdom of crowdsourcing of which I'm a prince, that is the focus of the vignettes that I'm going to tell you about in the following slides. But really, you can extract, you can disassemble this idea of the network of biothings into three components. First is going from PubMed into some sort of database, so populating this knowledge base and putting it into computable form. Second piece is building the knowledge base itself, and this is where Chun-Li Wu is going to talk a little about some of the tools we developed here, and clearly the EEI has many relevant resources in terms of infrastructure for housing biomedical data and knowledge. And then really applying that to some driving biological projects, and that's where I think the cardiovascular emphasis of our center really could give us some motivating problems. But for the purposes of this talk, we're going to focus on the first step here, how we're going to populate the knowledge base itself. You might think that the knowledge base can be, that process could be deconstructed into three steps. So first, finding mentions of the high-level concepts in the text, mapping those mentions to specific terms in ontology, and that would allow you to traverse the hierarchy knowing that hematologic cancers are relevant to these specific terms, tyrosine kinase inhibitors are relevant to these specific terms, and then finally identifying the relationships between those concepts. I'm going to focus on just the first step in this process, because there's actually a very nice gold standard that we can use to assess some of our methodology. So the corpus that we're going to use to do that assessment is a corpus developed by NCBI. Very simply, it is hundreds of PubMed abstracts where a dozen expert annotators,
over the course of the summer, looked at these abstracts and annotated disease mentions. Diseases is very broadly classified. It actually includes disease classes and symptoms and things like that. But nevertheless, it was a nice gold standard corpus that we could use to assess some of our crowdsourcing methodology. So the fundamental question is, that corpus that was generated by PhD professional annotators, could we replicate that corpus using a group of non-scientists, right, to collectively perform this same concept recognition task? We first tackled this with a system called Amazon Mechanical Turk. I'm going to give a very brief introduction to Amazon Mechanical Turk for those who aren't familiar with it. But very simply, it's a convenient way to get access to non-specialists to perform manual work. And the basic idea is we, as the requester, create various tasks that go into a giant queuing system that Amazon maintains. Amazon handles the interfaces between workers. There's, I think, over 1 million workers worldwide who sign up to perform these tasks. Amazon handles most of that interface, the billing, the payments, and things like that. They execute the tasks, and we get those the results back, and we aggregate them in some form, process which I'll describe in a little bit.
Andrew,
Yes?
why would the workers work?
For money.
Okay,
Yes, this is a paid microtasking site.
okay.
It gives us access to these work.
And Amazon pays it or you pay it?
We pay Amazon Amazon pays
Gotcha,
us.
gotcha. Okay.
Right. So, and Amazon, of course, takes a cut. So, that's how it goes. So again, the experimental design here identified disease mentions in 593 abstracts out of that disease corpus. We pay 6 cents per abstract. Amazon Mechanical Turk works on this idea of micropayments. You do something that can be done repetitively, very reasonably quickly, and you pay relatively small amounts of money. Each human intelligent task is to annotate 1 abstract from PubMed for disease mentions. And just to get redundancy, we show each abstract to 15 workers in our population. Okay? There's a lot of development that went into this, but given the time, I'm just going to cut straight to the chase and show you the results. Okay? So along the bottom axis here is our 15 workers, right? If, for example, we require that all 15 workers who saw 1 abstract agree that a given word or phrase is a disease, then what you see we have a highly precise result, right? If everybody agrees, most likely they're true. But you see low recall, meaning all 15 don't get every single mention, right? So at this end of the spectrum, you get high precision, low recall. If you take it where if any one worker in our crowd of 15 has a particular answer, gives you a particular answer, you get the opposite property. You get very high recall, meaning you get almost every single disease mention out of the abstract, but your precision is a little bit down. You get a lot of stuff that isn't diseases as well. And as you titrate essentially your workers at that threshold, you get this balance between recall and precision. The F-score shown in green is the balance between precision and recall. The maximum is at a K of 6. So if 6 people agree, we get the maximum balance between precision and recall at an F-score of 0.87. So this took, instead of 12 annotators over a summer, this took 9 days for a few $100. So it was a good way to generate and validate this, the crowd, right? A crowd of non-scientists being able to perform this biocuration task. So you might ask that, well, is 15 overkill, right? So what we did is we just subsampled that 15. Suppose we only gave it to 14 or 10, 13, 12, 11. And this is sort of along the y-axis, again, is the maximum F-score, and the x-axis is just subsampling fewer and fewer different people seeing each abstract. And what you can see is that it's pretty stable all the way through here. This is just a blow up of this region between 0.8 and 0.9. The biggest leap is between 2 and 3, where we get the greatest leap. And at about 6 or 7, you start hitting the knee of the curve there, where you aren't getting that much additional benefit. Just in terms of comparisons to text mining algorithms, just out of the box, two of them shown here, we get F-scores of 0.25, 0.35. So, our maximum F-score, this just shows the evolution of our mechanical Turk experiment as we learned how to take advantage of this resource. Again, we top out about 0.85, 0.87. You can see we do quite well relative to text mining algorithms. Comparison to human annotators, so back to the people who actually generated this corpus. If you just look at the agreement level between a single annotator and what eventually turned out to be the gold standard, you have an F-score of 0.76. After the annotators are allowed to talk to each other, to see each other's results, that gets the F-score up to 0.87. And I would argue this 0.87 is in some respects could be the theoretical maximum of what we might consider out of our crowd-based annotator. So we're pretty much at that theoretical maximum with our experiment with Amazon Mechanical Turk. So the take-home message here is an aggregate our work on Ensemble is faster and cheaper and just as accurate as a single expert annotator for this biocuration task of disease concept recognition. Again, we are just focusing on the first step in this experiment, but our ultimate plan and ambition is to push further down. I think this is the holy grail where there's even a bigger delta between what machine learning is able to do and what our human annotators are able to do. And you might imagine a scenario down the line where we're not just asking people to highlight diseases, but we're also asking them to highlight genes, and then we're also asking them to highlight essentially the relationship between genes and diseases. So I'm going to wrap up very quickly in terms of where another area that we're pushing this, right? So the question is, does the mechanical Turk scale? You imagine 1 million articles per year. Let's just say we want to do 10 annotators per article. Let's just say we get up to 4 tasks. We want gene recognition and drug recognition and relationships. Again, 6 cents per task. We're talking about $2.4 million per year to do all of PubMed. So I would love it if NIH decided to give us $2.4 million per year to just tackle this brute force, but I think that's unlikely. So our next strategy is actually to try to push this 6 cents down to 0. Okay? And how are we going to do that? Well, we actually want to engage the broader community of citizen scientists, people who want to be engaged in biomedical research to help us with this biocuration problem. So this is a website we launched, Mark to Cure. Again, in this site, we are actually, again, trying to replicate exactly the corpus that I just described. The motivation for these people is that they want to be involved in biomedical research. They want to contribute to the scientific enterprise. Eventually, we want to give them an avenue to contribute to research in a particular area that is relevant to them. So we're approaching disease foundations to be partners in this, and our response from Disease Foundation and patient groups has been overwhelmingly positive. Just some key stats quickly. We launched 3 weeks ago, actually, this experiment. We have this many unique users. We have over 5,000 documented annotations, 62% complete. This shows the tracking over time. And importantly, right, let me just show you some of the data from individual users. These were 5 of our most active users. These are their F-scores. So our most active users, this slide is actually from last week. So our most active users now have done over 200 different, but here you just show it about 180. And the quality is pretty good, although we do see some variance there as well. But as an ensemble, we're able to average that out. Importantly, right, and hitting that last point, as an ensemble, our citizen scientists are very accurate. This is, again, on the document that we completed. You see the exact same patterns that we see from our Amazon Mechanical Turk. So it gives us great confidence that our citizen scientists can be partners with us in assembling this network of biotech. Again, I look forward to brainstorming with everybody else here in terms of how we can contribute to this pipeline here. These are all the people that are involved. The team of Mark the Cure people is shown right here. Ben Good, who's been the leader and the brains behind much of this, is in the room as well. Other group members. Typically, I leave people on this slide, but for this talk, we actually asked people why they contribute to Market Secure, and this is some of our answers that we get, and I think it's just, again, gives us enthusiasm that people want to contribute to this process, and I just think it's particularly powerful. So, thank you very much.
Any questions for?
Sorry, are we going to do questions now, or
Yes.
okay?
Yeah.
Then,
Yeah. Are you doing this like study at home, or is it screensaver?
no, this is actually human labor. This is
But people are going to a website and then doing this. Would it be more efficient to have people have things sent out to people, like a screensaver?
well, the screensaver, I mean, because, you know, when there's screensavers on, they're not there at their computer.
Well,
Right?
yeah, but that's sort of concept, I guess.
Pushing it out, absolutely, and you know, a big part of citizen science is growing your community. So a lot of it is engaging, reminding people that we need their help, putting out focused problems that we think that are relevant to them. So, you know, I think the broader issue of how do we engage them through either attracting them to our site, pushing problems
Yeah,
to them.
for example, this person who's interested in cystic fibrosis, you could just throw cystic fibrosis papers.
That's exactly it. That's exactly it. Yes. And so we reached out to actually to a lot of rare disease foundations because the rare disease community, right, doesn't have a lot of resources and feels underserved by the research community. And so they're very motivated. In many cases, they're already digging very deep into the literature. And this provides a platform for them to have their efforts live on and contribute to a knowledge base and actually you know, aggregate the results of their community.
Yeah, a question is, what is the best use for annotation? Will you give the keywords to specialized search engine, to Google? What's your plan?
What are the tools?
What are the use? Now you get the annotation, papers get annotated, right?
Yeah.
So now how you could maximize the benefits of that?
Well, that goes back to, in my mind, the driving biological programs, right? So in the rare disease space, the one that we generically throw out is drug repurposing, right? Rare disease patients, right, often don't have drugs that are suitable for their condition, and the question is, of currently approved drugs, are the other ones that might have an effect based off of their symptoms and related pathways and things like that. I have an intuitive understanding or hope that this network of biothings would be applicable there, but really we're also looking for partners, you know, researchers who would be able to bridge that gap between a resource like the network of biothings and actually data mining strategies that would be able to do identify drug repurposing candidates or target ID or any number of applications.
But the regular guy, will he use PubMed or will he use Google to find those drugs?
Sure. And I think that's
because there are some issues there too, right? Some interesting problems.
Yeah.
I understand this to use the F score to benchmark the different, well, your tool. But I was wondering if you could comment on potentially certain applications that would benefit from maximizing recall a little bit more versus precision and actually balance. For instance, in the search where you don't want to miss too much, you can afford having actually managed throughout the Google does. If you could comment on that.
Yeah, actually, the best trade, or the current thought in terms of the trade-off is actually to try to use machine learning and text mining to maximize recall, and then use the crowd as a verification service, right? Where you show the sentence from which a finding was derived, and you just ask the humans, you know, here's the derived relationship, is it supported by this text? And there we use the crowd to really nail down the precision. That's the current hypothesis of the thinking.
So this addresses the vast information. It's already on PubMed, but how about all the stuff that's being put onto PubMed right now? Shouldn't there be a way that journals are participating in this?
Yes. So there's strong interest in the publishing, in the academic research community to encourage authors can put the knowledge that they're publishing in words in some sort of structured format. And ultimately, I think that's the long-term goal. But the publishing industry moves very slowly. There are powerful economic forces at work here, and I think that is certainly not on the horizon.
One last.
Are there advanced ways to reduce these contributions to the So you might have an expert or some reputation system in which someone is rated higher than the rest.
I don't really want to get into the business of assessing people's credentials. The beauty of the crowd is that everybody, feels like they're chipping away and doing their best. But it's very clear that different users have different properties, right? And so we should be able to take that into account. We haven't done that in a very sophisticated way yet, but that is something that we're hoping to explore.
Great. Thank you again, Andy.
One question. Yeah, so the beauty of we have multiple institution participation of the center is we're offering a variety of environment. I understand the crowd here has a variety of their education backgrounds and social economic status, and I think all of that contribute to probably to a certain extent variation of the data sets you're going to get back. In our case, both the UMFC and here at UCLA, we do have patient population that could be tapped into cross-source that Carol was talking about, as well as the education crowd, as our students are sort of having very fundamental knowledge in biology and pre-med or computational tools that they could contribute.
I think that's a great idea, and particularly the link to education. You can imagine is even I, as a scientist, read papers, right? Just having this catalog of relationships that I've read about it would be really valuable. So allowing people to store and browse that personal network, I think could be, with the links back to the primary research article, I think could be very valuable.
We have more time to discuss after the next topic.
So this time it's my pleasure to introduce Henning Kamjakov, who comes to us from Cambridge, England, where he's the Director of Proteomics Services, team leader at the EBI. Henning got his master's at the University of Bielefeld and came to EBI, or actually worked at the German National Center for Biotechnology, and then went to EBI in 1997 and has been there since, and really been a key leader in developing a number of resources, both for proteomics as well as biomodels, which is something that we've been interacting with, and as well as developing visualization tools. And so as such, I think it's fantastic that he's part of this center here at UCLA and has come this way, and I look forward to your time.
Thank you. And first of all, thanks to everybody involved for the chance to be here, to present here, and really to be part of this exciting program, especially, of course, to Dr. King, but also to many others whom I've met already over a couple of years in the collaboration here at UCLA. It's really a great fun to work with you. BD2K promotes the fair data concept, which has been developed by FOSS 11. The idea is data should be findable, accessible, interoperable, and reusable. I think Dr. Ping did a nice rephrasing of this concept in her introduction, which is our dream is to end the phase of fragmentation of proteomics data. And this is really a more concise phrasing of this approach. And this is what we set off to do quite a long time ago. We started in 2002, the activities in proteomics. And really then in 2010 we got really going with the preparing proteome exchange, which I'm going to present today. And at this time, there were lots of recommendations from different journals that you should really make your data accessible through depositing your results in a public repository, which at the time was not at all the case. So proteomics has not at all the good tradition of genomics, of where basically making your sequences is more that has been mandated from the very beginning of the field. And so it was not even a question of making your data findable, etc. The data was just not available. You would publish your results, but you wouldn't have make raw data accessible, at least in many cases. So the journals, to different degrees, encouraged it, but it was not really fully taken up by the community. But even if you were willing to be a good citizen and really allow others to benefit from your data, to assess your data, your conclusions based on the raw data, it was not really easy, because if you said, okay, I want to make my data which supports my publication accessible, then there was a question, okay, where? How do I do it? There were questions about submission. Which repository should I submit to? There were a number of repositories around. Should I submit to only one, the one which is my favorite one? Will people find it there? Should I submit to the same data to various repositories? Do I need to submit only the results or the protein identifications? Do I need to submit the raw data? From the user side, the sort of inverse questions, how do I find all the data sets which I'm interested in? How do I find all data sets, let's say, on chromatin? Do both pride and peptide atlas have this data set I'm interested in? Do they interpret it differently, or is it really just the same collection of files? And then there was something which was fairly specific to proteomics. We had around 2012 serious problems with repository stability. There were two major repositories in the domain, Peptidome, which closed in 2011, and Tranche, which was heavily used and quite popular, which became first unstable and then closed in the beginning of 2013, with actual loss of data which was published and referenced in publications. So this was kind of the worst case scenario which you can have in science, that there's published data which is then vanishing. And so the question was also, if I invest a lot of work into making my data accessible and going through perhaps multiple forms, documenting the data, will it actually remain publicly available, or will it just vanish? And so we set out to address this with the Proteome Exchange Consortium, where the concept is to provide only one entry point for the user. The user will provide the raw data, the results, and the metadata all in one go, in one submission process, to one of the participating repositories. In this case for proteomics, these are Pride at EBI, Massive at UCSD, and PESIL/Peptide Atlas at ISD in Seattle. But of course, in the log, we are also open to participation of additional repositories in the future. And once the deposition has been done to one of these repositories, and then the publication is out, then the data is released, and there's a message, an XML message sent to a central instance, Proteome Central, where all the data is then searchable based on only the metadata. So we are really implementing a distributed resource where only the metadata is centralized, and then you can access, you can choose what is relevant for you based on the metadata, and then you can access one of the supporting repositories for the full data sets and can retrieve it from there. And the idea is that we offer different views of the same data supporting different use cases. So resources like Uniprot and Exprot are only interested in a high abstraction level of the proteomics data. is this protein well supported by mass spec data? Is this modification, has this modification of the protein been seen? While proteomics repositories, other proteomics resources like GPMDB, which reprocesses raw data or say for peptide tablets, these need the access to the raw data. So that's why we want to capture all of the relevant data in one go. And then allow systematic reuse of proteomics data as it becomes available. Also in specific contexts, like for example implemented by COPA-KB, where COPA-KB is only interested in data sets which are relevant for the cardiovascular context. And because this is in the metadata, they can just have an automated filter or just manually look at what is coming out in terms of data sets which are relevant for us to include in our specialized context, so that you can actually have a sort of alert system. So this is the outline structure of Proteome Exchange, which we have set up since 2010 roughly. And this has really now become, in the proteomics domain, a kind of household name. And we've had rapidly increasing numbers of data depositions, going from 102 in 2012 to now something like we are processing about 4 depositions every single working day. And we've really increased the coverage of the published data, which becomes available in a systematic way through Proteome Exchange. As you can see here, it's really a broad spread of countries. Unsurprisingly, the US is leading in terms of depositions, in terms of experimental systems, species, interest, and also in terms of data volume. So there might be small, sometimes still 2D gel-based studies where the overall volume is probably a few hundred identifications. And then it goes up to huge studies. The biggest study we've had so far was a single study which deposited 5 terabytes in the database. And we had to really also work on the computational approaches, how do we transfer these amounts of data. We're actually relying on the specialized system as Era, which is up to 50 times faster than FTP and makes it possible to really transfer these huge amounts of data. And so the current overall volume in Proteome Exchange is around 71 terabytes. So we are really dealing with reasonably big data compared to the genomics guys. We are still small, but for proteomics we are really capturing a large amount of the data which is being generated. But still, the questions I listed earlier on remain. So will my data be there in 5 years? It's clear databases depend on continued funding. There were these programs I've mentioned already. In one case, tranche closure resulted in serious data loss. Peptidome was closed down in a very orderly manner, so it was properly announced by NCBI we are going to close the resource down. The data is still searchable on the FTP server, but it's also searchable in Pride now. As part of the Pride database, so we imported all of this data. But one of the major aims of Proteome Exchange is to provide stability. And that means, A, we try to be selective in terms of members of Proteome Exchange so that they have a strong track record in likely being there still in a while. But also, we are a key part of the commitment of joining Proteome Exchange is to sign up to mutual backup in case of funding loss. No resource is entirely protected against funding loss. Normally it won't be overnight, it will be something which you can see and prepare for. But all Proteome Exchange partners agree to doing their best to safeguard all the data in the, if one of the partners is expecting to lose funding. So, the data should be findable, the first of the FAIR principles, and this is what we achieved with Proteome Central. where you can, through a simple interface, search all the available data sets based on the metadata, the study description, the title, the instrumentation, the experimental system, but also some things which we can taught by experience. After a while of operating this, we added the name of the PI, not only the submitter, which is often somebody in the lab, but the PI as a search criterion, just because it makes it much easier to get an overview of what has been happening, what is the whole set of studies which came out of this PI's lab, and this has been a feature which has been appreciated. We also are about to introduce tags where you can distinguish between, let's say, technical data sets which are there for assessment of mass spec performance, where data sets which you might use as a mass spec operator to regularly get, for example, generated data from the same E. coli digestion or something like this over the years just to warm up your mass spec, and these can be very interesting data sets for technical purposes. But then, of course, you want to be able to put all things like, this is a real biomedically relevant data set, give me all of these, and then I will do my fine selection, or give me everything from the human proteome project. So once you've zoomed into which data sets you're interested in, you can get the more detailed description, which provides all the metadata. So the data is accessible in a common format, in a common framework. And once you've decided, yes, this is relevant, then you can zoom in and you can go directly to where the data, the actual data is stored in one of the participating repositories with this link via FTP or via URL. So you can also do this in an automated way and, let's say, automatically filter the metadata as it is being released, and then follow the links to all the interesting data sets and start reprocessing or something like this. Data should be interoperable. So that's the I of the packing principles. And proteomics has a huge challenge in terms of many different experimental approaches, which mean that the file formats being used in which the results are presented are hugely diverse, and it's very difficult to develop workflows which, for example, for integrating data sets work for many different data sets for different workflows. And so just to do a search across the proteome exchange data sets and say who has observed my favorite protein, what's my favorite modification, is very challenging. And so in the context of the UCO Proteomics Standards Initiative, over the years we have developed a set of standards for data representation which make that aspect easier. It doesn't make it trivial, but at least you usually now can much better, using these standards, focus on the science. So for example, the science of what Alexei mentioned, the science of integrating the data sets and the challenges which are associated with this, rather than just doing a lot of curl scripting to just match the data sets again, the data formats against each other. We are also collaborating with metabolomics increasingly on this aspect of developing standard formats, because at least in some of these, both for the representation of spectra, but also for the representation of final results, the challenges are very similar, and it's better for everybody if you can reuse the same file formats across multiple domains. So this is the interoperable aspect. And finally, data should be reusable. And in the beginning of 2014, I sort of reported on 2013 really was the year where data deposition took off, because we had this vast increase in deposited data sets. And 2014 really has been the year where data reuse took off. One of the two human proteome studies in Nature, which already was mentioned by Alexei, where, yes, there are different opinions on the quality of the study, but this study actually used more than 60% of the data was data which the authors integrated from other sources. So they really did systematic data reuse to a significant part from proteome exchange. In GTNDB, which systematically reprocesses data sets, proteomics data sets, 15 of the 20 data sets of the week in 2014 were based on proteome exchange data sets. And there's a number of other examples which really show that reuse, re-evaluation of data sets in proteomics is really getting into the conscience of the community and is really starting to happen now. And we are, of course, very happy to see this. So now, for proteomics, we have, I would say, quite successfully managed to group multiple repositories together to really form a stable collaboration of proteomics repositories. But it's only one domain. And of course, BD2K is about collaboration across many different domains. One of kind of the sister domain to proteomics is, as already mentioned, metabolomics. And there's a very parallel approach which has also been, and we've talked a lot between metabolomic exchange and proteome exchange. They're using a very similar data structure of distributed repositories. Oops, something went wrong here. It was not the update. Where there's a lot of repositories around the world. 4 are listed here, but actually the metabolomics workbench represents 6 different laboratories just in the United States. And again, they follow this approach of sharing the metadata in a central place, making it accessible, and then having the full data sets in the individual repositories. Another omics example is the European Genome Phenome Archive, which actually is being led by Justin Kaschal, who's in the audience that was talked later on. And this archive has another dimension. So this is really the EBI's archive for access protected data. So this archive exposes the metadata for each of the studies, which are there, but once you zoom in on what are the relevant studies for your interest, then it tells you how to access the relevant data access committee for ethics approval or other restrictions. So the metadata is fully exposed and accessible, but the data itself is protected. And so these 3 represent 3 different types of omics data. They represent different approaches of making the data accessible to the public. And so what we want to do as an early project, and we've just started on this in BD2K, is to integrate these and go from proteome exchange, coming back to the title, to multi-omics data discovery index, where we will develop an infrastructure for integrating data findability across all of these 3. The technical challenges are, I would say, relatively limited. We have to come up with a system which allows to integrate the different metadata records and make it searchable in one system. These are limited data volumes. It's not a very complicated data structure, so this is feasible. The big challenge is to come up with a common metadata representation so that we can actually, across these different types of data from different communities, identify what are the common data attributes, what are those which are frequent but not common, and make sure that we implement efficient search methods to really allow to identify the relevant data sets across all of these. So this is a significant challenge. But if we succeed, then this will be a demonstrator for findability across omics data. It will include at least 8 repositories in 2 continents and 6 different organizations. So it will really span a broad variety of data origins. It will integrate 2 different aspects, namely findability across open and controlled access repositories. And this is something which I consider very important also for proteomics metabolomics. In the genomics area, protected studies are almost standard now. Proteomics metabolomics is still very much used to the open access, which I think is important whenever it's possible. But for things like proteogenomics, we have to get used to dealing with data which is not fully open, and where we still have to learn how to maximize the community benefit of these studies through making it findable, reprocessable, in suitable systems with minimum overhead to get access to the data. But of course, keep the data safe. And of course, this will be a BD2K consortium collaboration with the Data Discovery Index project by BioCaddy. So it will be really across multiple branches of BD2K. And there's also discussions to bring this into a BD2K and BU Elixir collaboration. So that we really also see this, get this as a pilot for collaboration between BD2K and the Elixir program in Europe, which has similar aims like BD2K, but less funding. And with that, I would like to acknowledge all the proteome exchange partners, in particular Eric Deutsch in Seattle, Andy Jones, Liverpool, Leonard Martins in Ghent, Pierre Lavins in Geneva. So there's a lot of people who have contributed to this over the years. Juan Pablo Alvar, who was a major driver of the involvement on the Spanish side, and who unfortunately is not with us anymore. All the Pride team, journal editors who have been very supportive, collaborators, again, thanks to Pepe for allowing us to be part of this big program. EI collaborators with Steinbeck and Justin Pasha here, and of course, thanks to the funders for making all of this possible. And most importantly, thanks to all the data providers who make their data accessible so that others can benefit from it and do the classical stand on the shoulders of giants. Thank you.
Questions?
How will you update metadata?
So what we are doing at the moment is we simply send a new message which supersedes the old one, and because they are linked by the accession number, that's fairly trivial. It's really just deleting one file and replacing it with the next one in Proteome Central. Obviously, in the individual repository, you just do a database record update. But in Proteome Central, it's just superseding the new message, and it uses the same mechanism. So that means also that if you are, let's say, watching all of certain events to happen, you will also be alerted that something happened about this data set. We are also, this is a bit beyond here, but we are also working on establishing a systematic data flow for reprocessing. So you can take the data, reprocess it, for example, search for new modifications, and then if you deposit that data, it will automatically point back to the original source data in the spirit of credit attribution, so that you can really trace, aha, this actually goes back to another source data set.
So I assume you store the statistics on how many times the data set is downloaded or is partially downloaded, and would you be open to actually showing this information to each data set?
We're actually working on that. So I didn't include the slide, but we actually had this data generated already for our recent review. So for 2014, the highest downloaded data set was, I think, 525 times downloaded. And this is calculated as number of hits against all the files of that study divided by the number of files. So it's really only counting full downloads of the entire data set. Of course, if there's only partial download because somebody just wants to do a spot check, it's not even probably taken into account. But it's also clear that this is a typical distribution of, well, the data, anything that is in an impact journal raises a lot of interest and is downloaded a lot. And then there's a long tail of data sets which are probably download it once or not at all. But yes, we are planning to make at least the top X accessible for the statistics. We probably won't show it for everything, but we plan to show on the web pages those which are heavily downloaded. A bit similar to what we do in the DMC journals that frequently accessed. So it's also has a nice feedback to those who make that data available.
So that raises the question of that popularity is not necessarily an indicator of quality.
No.
Is there a way maybe for you to, for particularly high quality data set to be brought to the attention to people who are interested in mining data?
We wouldn't presume to be able to properly judge quality. I think this is something which is really the job of editors and reviewers. But what we are already, again, this will come out this year, there is a very simple data format called QCML, which allows to store a number of quality, or to provide a number of quality parameters for a data set. And we plan to automatically, on submission, generate, run a number of tests, which then will be presented, the results will be presented in this QCML and will be added to the submission. And of course, what we hope is that this will be seen also as a service by the journal, because it happens during the submission process, and it's kind of an independent assessment. So we are planning this. I don't know yet how well it will work, again, because it will only work for a fraction of the data sets because of the complexities of proteomics workflows. And especially when you are innovative and you provide a new workflow, which often is the highest impact applications, then standard methods of assessing this will not work. So where we can, we will do that.
I've been a big stick in this whole area of data repositories. I think this really should be a government function because, I mean, as was clearly evident with Tranche, and so it just kind of mind. And I thought the journals were pretty intuitive, and they're consistent with project data, but not moving resources up to do so. And they also were somewhat clueless to Ralph had a big argument, Ralph Brash on the city of the Tranche. He said it's perfectly fine because it's on the verge of collapse. And collapse wasn't the next one. And so he was just clueless about what the status was. But the thing about this is most captive, because I had lots of conversations with Dr. Sechi about that, something like that thing. And he and other people that and CBI to set them up. And then they shut down before anybody knew it was actually functional. That was just very annoying.
Well, I can only agree. I mean, it's a really sad story. It should never have happened like this. And I think it had a huge, especially Tranche, a huge damage to the support from the journals. I mean, NCP made submissions mandatory, and then the repository collapsed. So that's really the worst case scenario. And that's why we are emphasizing so much program exchange. It should be stable partners and stand in for each other. And we really feel we are sort of in a balancing act between not saying we are a club who can only play in this club when you are rich, and on the other hand, providing the stability. And all we can say is you need to have a decent track record. And of course, EBI has a sort of, EBI's mission is to provide stable resources.
Right.
So we have never shut anything
I can
down.
look at your American partners and I can almost predict when it fails.
That's why we say we will also stand in for each other. But we can also not just say it's, we only do one or two repositories. I mean, the whole point, and I think what has given Proteome Exchange the acceptance it has, is to be open and not just say, as pride, give everything to us, but say we want really to form a collaboration and we work together. And I think it works very well so far.
So, one conversation with Ralph and Rick about the stability of ISP.
Yeah, we
We
are.
can't see it sliding past week.
We are aware that it's not easy, but we are trying to sort of do as much as we can to provide this information. And ultimately, the best support of stability is if people A, use the repositories, and B, voice their support. Use it or lose it. In many cases.
All right. Thank you again to both speakers of the session. And do we have another break, or do we?
So, hey, your little quote up there, the open data release policy wasn't as open as you might have thought. I was at a malaria conference in Princeton, and a huge fight broke out. Some guy who was having malaria data, and the malaria sequencers were going berserk over that idea. They wanted to string the guy out by his feet because they felt that since they were doing sequencing, they should be the ones that published
If you're confident to produce then you are probably going to be a scientist, you're going to see right what
they successfully prepared a study published. Which I thought was not in the spirit of the open data share. I think
proteomics is on the way to improvement, making data available. So as much as possible, if we achieve what has happened in proteomics, in genomics, and that would be already much further than the past.
I think the data is much more complicated, so it's much more difficult to use to create value to other groups, because you're actually very specific kinds of questions.
Yes.
You see with the genome, you're just seeing the genome.
Yes, but we think that's an excuse not to make the data accessible. That's a bit like declaring your colleagues incompetent and yes,
It's a question of whether
All right, so how about we take this discussion offline? And, if we ever want to get to lunch, we should move on to the next session. Yeah, so first, I'm Jason Kong, faculty member in the computer science department. It's a pleasure to be in this BD2K team, and thanks Peter for inviting me to participate. And I have the pleasure to introduce the next speaker, Professor Carlo Zaniolo. He's also a member of the team. He and I have been colleagues in the computer science department for almost 25 years. And he has a distinguished career, first at Bell Labs and also at the MCC in Austin. Then he came back to UCLA where he did his master and a PhD degree. Come in as a chair professor in knowledge science, Carlo.
Thank you very much. I started my effort to see Windows. Okay. So perhaps let's hope for the better, right? Yeah, let's hope for the better. Okay. The graphics may be what they are, right? Okay, so basically this talk, or rather this work, was inspired by success in Wikipedia, which is, you all know what Wikipedia is, but it's a decorated web document corpus, which is being used by millions, over billions perhaps, and widely imitated by a lot of libraries and web, which specialize in scientific or special purpose document corpus, okay? Myself, of course, a couple of weeks ago, talking with Dr. Lin and Dr. Quinn, they told me that they were going to use some heavy water in some clinical studies, and I didn't remember much of heavy water except for the what World War II story about the Norwegian plant of heavy water. So I just first thing I did, of course, I went and I looked it up on Wikipedia, that's what I found, right? Immediately you can see the following that, to a large extent, the pages of Wikipedia are just like any other printed paper pages. They have an abstract, they have title, an abstract, a table of contents, and if you scroll down, you'll find references, etc., etc. But in fact, they have two new things which, are very important. One being the search box with which you can search using some text and keywords for relevant documents, and the other is the info box, okay, which is basically structured information taken from summarize some information pertaining to the subject matter. Okay, so that's what we are in fact going to talk about, because in fact what happened is that the Wikipedia people had this Wikipedia, okay, whereby they took the information from the info boxes, some template, they mined that information, and they store in a database or a knowledge base, which consists of triples, right, which can be queried using SparkQL, which is a structured query language, such as this SQL and other query languages that you use in database systems, right? And we'll talk more about that. But basically, using structural languages or a combination of structural language and text search, you get much better precision, okay? So in other words, you get exactly what you're looking for rather than a very large set of documents and pages, okay? Also, of course, there are other things in the Wikipedia, like an ontology, which is kind of simple but very effective. And that has led to a number of new applications, which often go under the name semantic web, right? In natural language processing, data integration, etc., etc. Okay. Having said that, you say, well, this would be an exciting idea to use in any scientific information system, okay? Particularly, medical science information system, okay? Which allow us more oriented to work traditional databases, which I've been working on for about 40 years, so perhaps they're kind of dated in terms of technology, okay? So, as we look at that, we realize that, in fact, there are some problems, okay? There are some problems, okay? And that's what swings our project, which is send for scientific web information management system, try to accomplish, provide those technical advances which are needed to basically extend this kind of technology to better support scientific information, particularly medical information systems, right? And what can we say about that? So what are the problems, okay? So why do we want to improve, okay? So great, there are a lot of problems, okay? And those limit the effectiveness of, the current technology. The first problem is usability, okay? Because SparkQL is a language for programming, basically, right? And even programmers, will have to put some effort, because it's not like the digital database where you have a schema which perhaps has 100 relations or something like that. You have thousands. So, and then there's an ontology, etc. So it's quite complicated, okay? So usability is a big problem, especially if you want to have, medicine for the masses, okay? Like, in fact, we are talking about. The other cause is that Wikipedia, yes, they did a reasonable job extracting information from the info boxes, but in fact, a lot of info boxes are not there, right? A lot of pages do not have an info box, or if you have, entries in the info box which are missing, certainly missing, or they are outdated or incorrect, okay? For instance, you look across different languages, you find that you don't have the same information, okay? You go to any conference and, they ask 5 people from different countries to look up their, their information about the local size, their own language, you will come out with different numbers, okay? For, what is the size of the pyramid, for instance, what is the distance between one place and another, they have different numbers, okay? Of course, Wikipedia is addressing that, okay? Now they're trying to reconcile all that. So, but it definitely is a challenge, right? So that's definitely an issue, right? And then, of course, provenance and paper, okay? One thing that you don't find easily now is who is the author of that piece of information? When was that put in? Was it verified? Was it audited? Okay, and of course beyond that, it's often the case that not only the time of the information was put in is an interest, but sometimes that particular entity has attributes which evolve over time, like and so you want to find the whole history, right, of that particular entity, and that would be the most informative thing. So that's the kind of thing that you would like to have. So you see there's a lot of interesting problem issues, particularly from the computer science viewpoint, and in fact we have tackled that problem in various ways. And for starting, usability, okay? Usability, as I said, it's only for programmer only. We have a new approach, which is that called, by example, structured query, which we aggregate into best queue, okay, just by chance, very convenient acronym. But in fact, it is a very powerful mechanism to class queue queries. And we'll choose the info box itself to enter the query. Of course, that will not necessarily be in the last step, because you still have to interact directly with the computer. Perhaps what you want, a natural interface. But that's also one thing that we will work on, but by preserving this best queue approach to ask query. We'll tell you more about this approach. In fact, if you want, we could even give you a demo about it. Okay? And then to complete an inconsistency, okay, what did we do? Okay, of course we didn't think about crowdsourcing, we didn't have resources for that. Basically we took an approach of integrating and realigning, because there's always some inconsistency which needs to be solved, existing knowledge bases, right? We did that. And beyond that, we said, well, a lot of information which is missing from the info box, it can be found by reading the text, okay, the page. So what you need a system which mines the text mines that page, and extract the relevant information. That's what I demand. Right? The third thing, of course, we did, we work with provenance, right? Or the fact that we plan to do, because this is still at the beginning. But, we want to keep the rich provenance information in the database, so whenever you get a page, the scientist gets the information, the scientist can already go back, who put in this information, okay? Because the authority of that information is very important. When the first encyclopedia came about, which was just before the American Revolution, at that point, there were people like Voltaire or Jean-Jacques Rousseau, okay, contributed to those entries. I don't think we can afford the same level of authority, okay? Nowadays, but let's put the name, we can say a lot whether in fact this was contributed by a famous scientist or some unknown person somewhere in some other country. So this is very important, okay? So that's what you have to say, right? So provenance is very important. And then, of course, history is very important. And in fact, we want to have an extension of the partial language, not only, but extension to the user-friendly interface with the language. And of course, temporal queries are very hard to support. You need specialized data structure underneath to support them. Correct? So all these are planned, and that's what they're going to accomplish in streams, giving up time and money, of course. And let me tell you an idea what the approach is. Again, in addition to a very nice acronym, we have also a nice interface. For instance, if you want to find a query, find all universities, okay, that's it's more than 1,000 faculty members established before then. What you do, you go to any university, okay? The one that, in this case, for instance, if you go to UCLA, why not, right? And UCLA info box up here, right? Now, the special button is a code swipe, which in fact shows that the same info box, but now the info box becomes active, right? So you can enter condition, right? Which is like this, okay? And this condition is shown there, right? You see those red fields, you have the condition like before 900, and then it's up to 1,000. You could have condition of public university, anything like that. But what's observed that there are two numbers up there, 900 and 1919, okay? And in fact, only the red one is that one, which is that's the only field that you can ask query, okay? Because you want to put in this single box, put additional new information. Like for instance, UCLA, 1919 was the time in which UCLA changed from a normal school to a southern branch of University of California, okay? Of course, that's not in the database, not in Wikipedia. So don't try to put a condition on that because that will not work. In other words, there's a lot of things that help the user. For instance, when it comes to date, you show particular format for the date, so the user knows that he or she should use that format to express the query. So there's a lot of system which goes wrong, okay? And then, of course, once this query is launched, the system simply return, what you finally normally, the answer, okay? Which is just the standard way that Wikipedia returns answer. There is something new, for instance, there is this summary, there is also an info box for the answer itself, okay, which summarizes, how the query that you launch, also allow you to put additional conditions, there is this property, property relational system, which is supported here, and so on, so on, there is more richness and more possibility, okay? And so in other words, a very powerful and rich system really address the usability problem in a very effective way, okay? But, okay, that's the structured part. But you remember the info box, okay, and on top of that, we have the search box. The search box is still there. So any condition that you cannot put in the info box, you can put in the search box, okay? And the system will search for both, the combination of the two. And guess what, okay? So basically what happens is the combination of two returns, better than shown, returns out a much better precision, okay? Precision than just this text or condition, okay? Remember the precision is the name of the game here, okay? Because search engines return a lot of results, okay? And as Dr. Chu was saying, there are papers published every day at the incredible rate, so it's been more and more results, okay? Then, and that was the problem with the search engine at the beginning. Then Google came along and solved the problem by showing the most important result in the first page, okay? Oh, and that made, okay, let's go. And then made the comment here, we want to address that. And let's continue because we have a time constraint. And, so these are longer term goals. Natural language fresh answering, okay. Support for powerful analytics, okay, that's another thing we are adding. More powerful analytics because those will lead to, OLAP will lead to KD, knowledge discovery, which are very important. So we'll get, the gain on knowledge discovery from that. Okay. So we are only halfway to my presentation, thank you. And basically what I'm saying that the fact that now we have high quality, high precision search engine shows the imperfection of the underlying database, and so we have to address that problem. How to address the problem? Well, let me give you an example of this problem using ratio McAdams, right? I don't know why ratio McAdams, but anyway, if you go to, you find, for instance, inconsistencies, like there are two possible dates, okay, which were corrected, and then sometimes you have things like, sometimes you have things like the information, the text, which is not in the info box, like the award that she got, okay? So really what we're doing, okay, we want to have information which is complete, accurate, up to date, okay? That's essential in making information system. Complete, accurate, up to date, all right? How do we do that? Well, there are a number of approaches, particularly crowdsourcing approach. But what we did, we started by integrating a line databases, text mining, knowledge from document, and finally manage provenance information supporting authorship and time queries. That's what's happening now. So what we do, an integration for to put information from various databases, and then what? And then, okay, we mine the text of Wikipedia, we got more information. How do we mine the text? Well, using basically standard for natural language parser, whereby you take a sentence like ratios Canadian actors, and say, okay, there are information ratio is an active ratio is Canadian, but that's not enough to knowledge base. What you need to do, ratio of occupation is an active, the ratio nationality is Canadian, right? Now it starts making sense. You can put that in a triple format. You can query that, etc., right? Okay. At the end, when you have done everything that done, okay, with all this tool, you always need some human information, okay, to validate or improve the result obtained in terms of correctness and significant elements. In fact, we have something which is known as the infobox editor, and which looks like here, it's still very primitive, but anyway, it can tell you things like, okay, for instance, you want to give feedback on the entry, that's something that you can go to swipe to get more relevant information, okay. Sometimes what you have all of these synonyms or homonyms, right? Like, typical problem is board. A board in the text may mean the place where the person is born, or the place or the date where the person is born. And there are some statistics which will help you resolve what's the meaning, okay? And then, of course, one thing that we have a ranking, okay, and we have also some provenance. Now, the only information about provenance that we got this from Wikipedia, the one of Yahoo 2, we got something for our IP miner, okay, much more information. Okay. You need information about when was the information entered, when was it audited, okay, etc., etc., all right? And by whom, by which organization, etc., etc. Who was the PI? Remember the previous speaker said exactly that. Okay, let's go back because it's about time to talk. So both better provenance support is needed, author, editor, and time. Okay, and so we need to support the queries, which not only mean the extension of the query language, but also extension of user interface and efficient implementation of that. Okay, these are the research teams which are working on. And now, guess what? How many minutes do I have left? Okay, fine. Conclusion. So the implementation of repeat and scientific information system are well recognized. The medical scientific application require much better usability and quality. Okay, those are the two things. And the students have been on our framework be able to tackle the problem by making much easier knowledge retrieval by query express with the best view interface and also natural language interface built on this best view. Okay, it's not just a straight natural language interface, it's built on that. A much higher quality by text mining the pages, data being aligned knowledge base across the knowledge base, support for provenance management and auditing, and high precision of query answer, which combine the structured query with text search. Okay, so that's what we have done, and those are very, very ambitious goals that we have done. That we are doing. Okay, some of these goals, like probably be achieved, like as I said, we give a demo, but some other are in the work, and for that we would like to acknowledge the people who have done their actual work, and I would like to thank them, and hopefully they will be some of the papers that you will reference, and and finally I would like to thank you for visiting my presentation.
Carl, if you don't mind, we have a panel coming up the next talk, so we can collect the questions together.
Okay, so we will sit down.
Yeah, you can sit down. We can bring another speaker under that, and we can prepare a panel for the discussion. So the next one is coming from the guys going to tell us more about the database.
Hello everyone, thank you for the chance to talk today and the chance to participate in this interesting effort. I've already had some interesting conversations about this and other topics with people in the audience. So one of the projects in my team at EGI is the European Genome Archive. The mission of EGA is to enable collaboration over data types which are meant to be consistent in a way that can be shared among scientists, but not completely openly. They can't be put on the internet for anybody to download. They have to be subject to a controlled access. And so I want to start out and say that this is not a problem that is necessarily unique to genomics. I know that there are data sets for ethical or clinical reasons, the way that the data was obtained in research study, where even proteomic data is subject to having to go through an approval or IRB process by which individuals need to apply for access to get to the data. And the EGA is a generic mechanism by which you can have a platform to share that data and to manage the permissions over data sets. It's also relevant in the sense that more and more data is being captured on the same cohort patients at different levels. So even if the proteomic data was considered to be sufficiently anonymous to be public, we want a mechanism to link that proteomic data to metabolomics, to clinical phenotypes, and to genomic data of the same patients. And I think we're going to find from a systems biology perspective that when you can layer those many different kinds of omics together, the results are more powerful, but you need then a secure mechanism to do that. Because obviously genomic data is going to be traceable to a single individual, and increasingly an individual genome is going to be available through various ancestry sites, not on the internet. So that data is something that needs to be protected. So what is controlled access? I just want to walk through sort of the scenario of how you would use the EGA. And again, the EGA is a resource hosted at the EI, or the EDL organization in Europe, but it is a global resource. We have data sets from all over. Users discover the data set either on our website or in the future through one of these cross data set registries, as Henry mentioned. They discover the data set exists, but then they discover that they can't just download the data, they need to go through a process. And so the first key is to inform users of the existence of the data set and to show them the steps they have to jump through to get access. That request is then reviewed by a data access committee, and then the EGA provides a set of web admin tools for the committee who reviews the request to go ahead and grant access to a given group. And then once access is granted, then we have mechanisms to deliver that data to the authorized investigator. Right now, this is through secure data download, but as I'll talk about at the end, we're increasingly moving towards a model where bringing the user to the data and allow them to access the data in a secure cloud. And one of the purposes of that secure cloud would be then to integrate both the genomic data with the proteomic and metabolomics. To give you an overview of what is in the EGA now, we have more than 450 studies available for requests. If you're familiar with next-generation genomic sequencing, this is often in the form of these BAM alignment files. We use about 30 terabytes per month of submissions. Again, we cover not just genomic sequencing, but next-generation sequencing-based RNA expression profiling, methylation, and we can certainly cover proteomics data and plan to for total opinion. The review covers everything from rare disease, genetics, cardiovascular, cancer genetics. And we have perhaps a pilot of this metadata exchange system with the NCBI, and you can go to our sister resource at the NCBI, and although you can't get to the same raw data at both places because it's controlled access, you can discover the existence of the data set in either place as if it was one federated system, and I think that idea is expanded on by the exchanges that Henry spoke about. So there are 4 areas of focus with these things. The first being patients, that we want to inspect the terms on which research data was collected based on the way the patients consented. Often an overlooked category here, the ethicists will often talk about the concerns about privacy, but another angle that someone also talks about is the patient's right to share the data. If you talk to somebody with a rare disease or somebody with a family history of cardiovascular disease, often they don't want to be restricted from the ability to share their data for the benefit of others. They need a platform to be able to do so. We want to respect the interests of submitting investigators. Often one of the big concerns with the submission of data to centralized archives is the need to publish, the need to have sort of first rights on a given data set. And so we have many mechanisms at the EGA to allow data to be held privately for a period of time, of course released. Really the ideal scenario of these kind of archives is to use it as a means of supplying supplemental information with publications. So we'll submit at the time the data is generated, we'll hold the data until the point where the first submission is available, and then we'll release along with the publication. Although there are advantages to pre-publication release as well, we encourage that whenever possible. We enable the work of other biomedical researchers and methods developers by making these large data sets available at the raw data level to groups other than those that produced the data originally. And finally, we look to thwart the interests of funders and ultimately the public taxpayers who often fund the creation of these data sets by allowing data sets to have a life far beyond the initial study in which they were generated. One important issue with the way that the data access control is managed at EGA. Often, when they first hear about this kind of resources, people think that we have a committee meeting in a dark room deciding on who has access to what data set. And it's for the Scrum, the EGA works very hard to provide transparency and really put the data access control in the hands of whoever it is that owns the data, the funders or the submitters. We just provide a mechanism for them to enact the data access decisions to those groups to make. So to get an overview of what it's like to interact with EGA, this is our front page. There are several different angles you can come at to learn about study. You can search based on a phenotype, cardiovascular disease, neuromuscular disease, cancer. Specific types of cancer up here. If you knew a bit about the existence of a study, and you can search by the name of the study or the name of the cohort. An example data set is the BD2K study, which is 4,000 samples with 6x whole genome coverage and a bunch of detailed exome data. This is a very interesting study. It looked at extreme phenotypes, so extremes of obesity, extremes of BMI, some rare diseases. And did deep genome sequencing. And importantly, they did a large control cohort at the same time. So that is another point. Often the value of these archives again is to provide a large control population. So if you have a focus study, you can easily gain access to a large control set and then do a much more powerful statistical study. And so you discover the existence of a study you wanted, you invite that study and say, hey, this is useful in my research. You click through one of these data and you would learn, oh my, we don't have access to download. And in a perfect world, I would like as much data to be completely open and public domain as possible. But again, this is data that if a system like this didn't exist, it simply would not be available beyond the lab in which it was generated. So although it's bad news, it's not that bad of news because we provide a link to find out about what are the steps that need to be taken to gain access to the study. And you click that, follow it, and you would then learn about specifics of the BD2K. And again, this is the wall between the EGA as a platform and then the specific study. We link you to the study-specific website where they can get credit, and also they can provide whatever study-specific rules there are about how to gain access. So you will learn there that you need to fill out this form, and you need to send it to a specific place. That's again in the hands of the study. But then the group that manages reading these applications uses the EGA Common Web Interface or a API which they can integrate into their local work chain to then go in and grant access to the study. So once the requester has been granted access, they log back into our system, log in with their username and password, and they find that they are granted access and they can download data in these formats. You can request to download data. And then this uses 7 different mechanisms, FTP, or the very large data sets as Henny mentioned, things like Aspera or something called Grid FTP, which can be used to transport literally terabytes of data. But in the end, we found that we weren't really enabling quite the broad use of this kind of data that we hoped, because in fact, the groups of labs that have access to hundreds of terabytes of data store and the compute clouds to work on them, often were the people that already have a lot of data anyway. We were wanting to reach a larger audience, smaller labs, smaller groups, new groups that didn't have necessarily that much infrastructure. So a big focus now is to provide cloud-based access to this data so that you can log into our systems securely, have your own workspace, and work on the data there. And it's also a good preview tool because even if you have a local infrastructure, it's going to take weeks or maybe even months to transfer large data sets. So it's very useful to have a sandbox where you can get these data sets in an interactive cloud environment to play with them, do some initial analysis, prove to yourself it's really useful, and then if you really need access to all the raw data, then you can go about downloading terabytes to your local system. I see at this point at the beginning here, I'm going to work with hinting on the metadata discovery portal. Another point is secure APIs, and this is in coordination with the Global Alliance for Performance and Health. I'll talk about it in a second. But then, again, this is that I usually open up a live, real-time interactive stream with the data store you're looking at. Even in some cases, have your algorithm working on the data remotely as if it was on your local machine through a REST API. Because many types of analysis are not going to require that you download all of the data. You need slices of the data, and you need it to be delivered through a secure connection. So open up an HTTPS connection, do your REST API data, and you can interact with data in a remote fashion. And you can get over that sort of barrier of local infrastructure. So then, sort of a graphical representation of this cloud model of interaction. You have the data generation up here, you have the users down here. So there's two ways you can allow the user to interact with the data. One is moving the data to the users, so they can again do this through download from the traditional archives and CPI or BD2K, for example. And another perhaps more modern way of interacting with remote data would be through interactive REST APIs where you only download the data that you need at a given time. But if you do need access to large core of the data all at once, then it may be better to move users to the data. And so then we have a local private cloud at BD2K, also other partners at CSC in Finland and in Barcelona, and potentially any others that we could form an agreement with and make sure they conform to the same sets of standard. So to do this, and especially to do this in a way that gives controlled access data, there has to be a set of core services. We are working to establish those core services not just through EDI, but with the Elixir network in total. It would be good to work with BD2K on this type of issue. These are just the basic mundane problems of who are you? How can you establish the authentication of a person? How you track what a given individual is authorized to see in terms of data sets. So tracking authentication, authorization, tracking a central data set registry, having a stable hub for that data is something we need centralized infrastructure to do. In Europe, that will looks like it's largely going to be under the banner of Elixir for now, and it sounds like that BD2K and Elixir are going to have a useful collaboration in that space of sort of framework infrastructure that you can build on top to enable these federated and remote services, which require that you track individuals so that you can give access to secure data sets. Just a little bit more about Elixir. I'll be interested to hear more about what we can do between Elixir and BD2K. And finally, this effort called the Global Alliance for Genomics and Health. I would be interested to know if any of you, especially in proteomics space, are familiar with the Global Alliance. It essentially styles itself as being the W3C internet standards body, but for the area of genomics. And so they're looking at things like standardizing API calls, standardizing data formats. It could use a proteomics angle to it. And that sounds like a lot of the work that's already been done could be folded into sort of a larger view of GA4PH. So my acknowledgments of the EGA staff, we're so happy to be part of the BD2K team, a number of EU grants, and the EDI whole funding. Thank you.
So at this time, I think we're going to do the panel discussion. The panel is roughly around the idea of building tools, but certainly I think before you go, Justin and Carlo, why don't we, there's also the opportunity to just ask questions of these two previous speakers. So people can throw to read. I'm sorry, yes, only very right ones.
Yeah, you can get in front.
Yeah.
So Justin, can you say a little bit how big is your cloud? You have one part of the data depository place, right? The other one is the cloud that allows you to do the computation.
So how big is, I mean, I say how big is the server of metrics? I mean, it has access to a, 4 petabyte data store. So I mean, the total storage is large. The total number of compute cores, I don't have it off the top of my head, but I mean, it'd be on the order of thousands.
I guess if you want to do an association study or something, like how scalable is the computation.
Sure. So I mean, we have substantial resources, and we're looking at ways to have sort of an elastic cloud kind of thing, so that you could dig into resources which weren't always allocated. And you definitely don't want an individual group to freeze a set of resources. You want it to stay on track. I think the real solution to this, though, will be what they call cloud bursting between different clouds. So I listed the BSC and the CSC. Also, I've been working with Chicago, Rob Rosen, who has a lot of biounits, I think it's called. It's a large kind of biomedical cloud. So I think you'll get the amount of resources you need only if you can really distribute it globally. Yes.
Okay, the same question which Alexei asked me. So, but you have a better handle on knowing who is reusing a data set, because I have to ask for access if I want to work with a data set. So you already know I'm a candidate who might be publishing on this data set. So do you track and do you provide, for example, in the sense of associated publications, who has used the data set? Is that available or is that planned?
I don't think we have. I think for privacy kind of reasons, we don't have a public tabulation of who used each data set. I think it would be up to data set owners if they wanted to have such a sort of registration. I think we have the capability to make such a list if the list desired.
So maybe I'll ask you a question and bring it back to Carlos' talk. One of the real interesting parts of the integration of various community knowledge bases and bringing that together. One of the unique things about Wikidata, as we talked a little bit about yesterday, right, is the fact that it is meant to be a community knowledge base. So what's your input on essentially feeding that back into Wikidata?
Well, we are coming from a technician viewpoint, so, but basically that's the part which excited me too. Okay? The database and the fact that also you will use user-friendly tools for people to access. Okay. Basically, we have to learn from Facebook, right? There's a tremendous potential in the community. When people find it fun to communicate, to contribute, right? To talk. And, it's not, so our challenge is to provide those kind of tools to the public general, particularly to the scientists, to our graduate students, so they really become a very lively community, okay? And I think you're doing just the right thing in that framework, right? And, I think this project, remember, being together, we didn't know about the challenge of this project, okay? And it seems like you were doing some very similar kind of work and not very distant from each other, okay? Without knowing that. That's why we need a community to change all that, okay? People who have the contribution which dovetail with each other should in fact communicate, okay? And this kind of tools that you suggested, working out its, sociological potential, basically, is very important.
May I add a comment, actually? I think one type of data which will be interesting to see if it exists going forward is the space between clinical HIPAA-type data stores and completely public data. Because that's what really this is trying to serve here. I mean, the real HIPAA data is going to be very locked down and pretty difficult to get to. And the completely public data is completely free. What we try to hit here is a middle ground where provide a acceptable level of protection, but then still allow data to be freely used by bona fide researchers in that space. So really, to some extent, the need for this resource will depend on whether or not that is a substantial category of data. But I hope it is, because I think that...
I have a general question, Mark. Okay, whenever you hear about physicians learning from past experience, I heard that they got data from Holland, from Norway, they never tell me that they got data, clinical data from the US. What's stopping? Why, for instance, you get schizophrenia, you go to Holland, you go dental data, you go to, I think it's Sweden, right? Because they have kept databases of their experience. It's something, since I'm an outsider, I can ask that question, to be honest. Something wrong with the NIH? Was that wrong with the structure which doesn't make the same kind of information available to the public? Anybody care to take on that question?
So I think the VA is one example of in the U.S. where they
Okay,
have,
good. I'm glad. That's an exception.
but it's, I think, because we haven't had medical records and we haven't had as much infrastructure as there has been in Europe, before, then I think that's one, but we're catching up.
Yeah, catching up. I think that's probably the way to go. I mean, because over time, that will lead, those things will pay off very well over the long range, right? Yeah.
It also has to do with the single payer versus multi-payer models within the country. So countries that have single payer models sort of have a consolidation of data, whereas in the U.S. it's spread across a number of different insurance companies and institutions who are gathering the data. So it's much more difficult to get sort of the population-wide data sets.
Statistically, one sanitary data should be made public about implementation. Well, hopefully that's difficult to
I think in the U.S. there's a very strong sense of privacy that
when the privacy people were passed away 10 years ago, and nobody knows, okay? I mean,
and the flip side is actually something that Justin alluded to, right? At one point you said, talking about patients who are interested in researchers making maximal use of their data, and they're anecdotally you certainly hear people who are suffering from disease, right? Actually, the privacy issues are much less important to them. So I wonder, on your perspective, or anybody's perspective, in terms of how far you think we can push that in terms of enabling patients to even allow their data to be used more broadly.
So I'm really sad that Google Health doesn't get this anymore, but there's sites like that, something called Patients Like Me, where people share in a very social media kind of way. If those data can be made rich enough and semantically mineable, it's very useful.
Yeah, ultimately it comes back to the patient consent. So, developing consent mechanisms that allow patients to really understand what the challenges and the risks might be, and give them that choice. And then once you have the consent in place to more broadly share it, makes it easier for researchers to sort of carry out the work.
So since you guys have done a lot of work organizing data, so certainly efforts like the Personal Genome Project have gone to one end of the extreme where it's like everything's out there. And then status quo is very much, HIPAA, privacy, yada, yada, yada. Have there been large efforts to explore the spectrum in between?
Yeah, my colleague John Wilbanks in particular is one person who's looked at this quite in depth, and we're in the process of putting out what we call a participant-centered toolkit for consent, which is really putting the focus on the participant and less about the regulatory hurdles that are in between a patient, a researcher, and the work that's being done. And we're not the only ones trying to do this. There are many, many groups. Patients Like Me is another one. Susan Love and her Arguing Women, they have a process of work as well. So I think the people who are highly motivated in this space tend to be these foundations, the patient-centered organizations who are really interested in pushing it forward.
So in the same way, have there ever been, or is it even realistic to go for an overall sort of framework, consent scheme, where you might have different modules, but it would be much more compatible between different studies, and again, probably anecdotally, probably patients would be in many cases happy to say, yes, I give more or less full consent, but also then there's a subgroup where you 1071. This task of restricted. So, let's say, will it be UK nationwide?
So there are a couple of, so the GH, 4G, global lines have their, because they consent and governance portion is very specific to genomic data, and they're trying to standardize it there. I'm actually chair of a working group coming out of the White House OSTP, where we're looking at exactly that modular consent. And the modular consent can be based on the data types that are being collected, not on the research that's being carried out. And so the consent is much more based on the risks that would be associated with collecting certain types of data. And the hope is that that would get put out as a community resource such that it would be like a Creative Commons style chooser where you'd say, I have these types of data that are in my study, building a framework of consent that would allow for broad sharing and reuse of those data.
So going forward is much different than going backwards.
Absolutely.
And so stuff like the Jackson Heart study, it wasn't until visit 2 where this whole concept of having this broad shareability came into being. And so, you know, sometimes you want to have access to those samples, but you have to go back and re-consent the patient, re-consent the subject. And usually those, like Framingham, et cetera, are set up to just hear that we're going to bother the subject minimally.
Yeah.
And so, you know, there's that. And so going backwards is much harder than forward.
Okay, so this is an issue of, privacy interpretation. That issue is, even data, they are information, they are, how you find it, right? And that's sort of the back to the question that posed you, okay? So, what's the best, how are you going to place the search capability search engine? For instance, some community specialized in certain diseases, certain kind of disease, are you going to give that to Google, to Wikipedia, to Hangout, so to make it very widely accessible? That's another issue, right? I don't know if anybody is over, right? I mean, you could say, okay, the centers manage that, but do they really have an incentive or whatever, right? To do that. So, what do you think you should give that to Google?
Well, I think certainly.
Google is just another sector, old sector.
Sure. I think data warehouses and data repositories, like the one Justin's talking about, right, that are privacy aware, right? In an ideal world, would be able to respect the spectrum of different data sharing mandates from the data owners.
So the question is, do you think about specialized search engine, specialized provider search capabilities, right? We think you put it back as a general framework, which, Google is great. In fact, this announcement that you're going to support this is so they are great. Okay, but so why don't we give data to information to Google? What's our plan there? I mean, you must have a plan, right? That's what I'm saying, right? You see what I'm saying? We want to enable the people to access it, but how is the best way to go about it, right?
So I would hope that if it's a basic vocabulary of disease terms and consent categories and way to name the word a subject, which is name the word patient. If the basic vocabulary is defined, I would hope that it wouldn't matter if you could represent the XML to RDF very generically, you can index it in multiple search engines, multiple mechanisms. You wouldn't have to decide on which one.
I want to say something very similar. Solutions like EGA or what we built with Synapse. I don't think there needs to be one of them, but these data repositories that have metadata that can be indexed by a place like Google, like other search engines, make it discoverable, but having the right privacy and governance structures in place to ensure that the underlying data, what is truly sensitive, is maintained.
Well, there's more than that, because the one thing is for a person sick to look for a cure disease, another thing for, some company, to look for those people so they can hit them with intense advertisement, which is a great nuisance, right? saying? Yeah, all right, misusing, okay, so there is some risk there too, I mean, there is some risk with the art, this is a problem, right? Other thoughts, questions? Great, thank our two speakers.
I think, Jennifer, we're going to just launch right into the next topic.
No, I was just setting up earlier. We have the next planner.
So we're just going to launch right into the question.
I thought you said lunch. There are a lot of different accents in this program.
Try to keep my California accent down. It's my pleasure to give a very brief introduction for John. I think John doesn't need much introduction for this crowd. John did his PhD at the University of Virginia with Don Hunt. After a postdoc at Caltech, he moved up to be a faculty member at the University of Washington. In 2000, he moved down to sunny San Diego at the Scripps Research Institute. So throughout John's entire career, John's career has been characterized by really innovative work in the field of proteomics and mass spectrometry. He's regularly cited in the list of most cited scientists, and I think it's a real pleasure and honor to have him associated with our center. So without further ado, John.
Thank you, Andrew. I got kind of a general title bestowed upon me for my talk, so I will just run with it. This is our computing facilities at Scripps. But funding's been a little bit tight. This is, so I've been reading Walter Isaacson's latest book, The Innovators, and they talk about the birth of computing and the internet and all that stuff. And this is the ENIAC computer. And what's interesting about it, which I thought was a relevant point here, is that it was built by men. Men developed, designed and building. It was programmed by women. And there are a group of 5 women that were involved in programming this thing, and there was a team led by Grace Hopper, who's also a female. And so really, the first software engineers were all women programming this thing. I think this was one of the first programming computers. I also learned in this book where the title for the movie about Alan Turing came from, The Imitation Game, without having to go see the movie, which I thought was good. So mass spectrometry has evolved quite vigorously, or quite nicely from this stage. This is Thomson's cathode ray tube with a little magnet in it that bends the beam down to here, going through a number of different phases. But it's a technology that has evolved alongside a number of other areas, particularly the electronics field. These are really just now great big huge computing devices that have some hardware in them to move ions around. So what I thought I would do is since this is based on this talk spoke about analytical chemistry and computation, I'll talk a little bit about the innovations that led to these large-scale proteomic methods, because really it's very tied into the fact that we're now creating big data. We have to analyze that data, and we have to get knowledge out of that data. And just to show you that mass spectrometry, proteomics, mass spectrometry of peptides and proteins wasn't invented when T.S. Mann got interested in it. It goes all the way back to 1959, the year after I was born, where Klaus Biemann published one of the early papers on this idea of using mass spectrometry to do structures of peptides and proteins. And what really set the field in motion was in 1987 there was 2 nice ionization techniques. Ionization is the horse that pulls the cart for mass spectrometry. But what was most important with electrospray ionization is that now we could attach liquid separation devices to the mass spectrometer, which is something people have been trying to do for ever in the field, and it really didn't come to fruition until John Fenn
invented the electrospray ionization technique. Although that was Malcolm Dole that showed that you could get ions out of electrospray. This was such a huge invention development that Nobel Prize in Chemistry was given out in 2002 for this, for MALDI and then for electrospray. You've already seen this slide. This is the original incarnation that was made, I think, 25 years ago, and it's circulated around a little bit. But the second revolution here was tandem mass spectrometry, where you have two mass analyzers, and they can be used in different ways. You can use it to separate ions, to select out a mass to charge ratio. You can then activate those ions to fragment, and then you use the last mass analyzer as a way to sort those fragment ions. Most mass spectrometers today have a lot of components of quadrupoles in them, quadrupoles, octopoles, and so forth, and all that's developed by Wolfgang Paul. He published a theory, the quadrupole theory, back in the late 1950s, 58 or 59 or so. Hans Damelt was involved in the construction of ion traps. And note these were such important developments that the Nobel Prize in Physics in 1989 was given out for their efforts. This is probably about $1 billion a year business nowadays, so certainly a nice award. So all the techniques that I'm going to tell you about were really sort of described in this paper in 1986 by Don Hunt's laboratory. And although we weren't using electrospray ionization, we were sequencing peptides by tandem mass spectrometry, and all the sort of strategies that were developed, described in this paper, still exist, and it's the way we do things now, with the exception of the way we interpret the data, because back then, data was interpreted all by hand, and you know that when peptides fragment, they fragment this way, they fragment that way, and you get adjacent fragment ions. When you look at the mass difference between adjacent fragment ions, you can read off the sequence. What makes it complicated is that you can't tell the difference between ions, between this set of ions or this set of ions in the mass spectrum. Although these are nicely color-coded, you can't tell, you really don't know that this is an ion from the N terminus of the peptide and this is an ion from the C terminus of the peptide. So it makes the interpretation of these things actually rather difficult. Okay, so the big unexpected game changer, of course, was protein biochemistry. For protein biochemistry was the fact that genomes were being sequenced, this data was being deposited. As Henning pointed out, it was very nice that the genome community was putting it right into the database so that we could use it right away. And that was helpful, although it's not really until a genome is completed that it really becomes much more interesting and useful. And a big week, one of the things that we started on was to sequence all the ribosomal proteins out of yeast when that genome was done. And one of the big problems that we had in doing that was that the nomenclature at that time was all erratic, and so we really couldn't figure out, did we get everything, do we have everything? Because there was some of the ribosomal proteins were named using the rat nomenclature, and some were named with yeast nomenclature. So it was very confusing just because of nomenclature alone. Okay, so the third piece of the puzzle for proteomics was this idea that you could use these genome sequence information to do fast and accurate lookup of information, just like scanning the barcode. Now we're scanning tandem mass spectra of peptides, and we can get out information about where those peptides came from, and hopefully information about proteins on function and so forth. That all can be done very, very fast. And so this convergence of technologies in this time frame of improved mass spectrometers driven by the fact that we can now ionize biological molecules much easier, sequences, computational algorithms, because everybody had personal computers. I had a personal computer on my desk when I was in graduate school that I was able to buy with very little money, $1,500, which was a lot of money for a graduate student back in the late 1980s. And so out of that has really emerged this idea of doing proteomics. But there were several other innovations that were necessary to really drive us to the point where we are today, and not the least of which was electrospray, which allowed us to integrate liquid separations and mass spec, but also data-dependent data acquisition, capillary chromatography, and multidimensional separations. This idea of doing data-dependent data acquisition really grew out of this idea of instrument control language, and this was developed at Finnegan, which is now, they're now Thermo, and there was a couple of engineers, particularly this guy, Urs Steiner, that came up with this idea of not only just using a computer to acquire the data, but also putting computers on the mass spectrometer to control the operation of the mass spectrometer in real time.
And that was really a nice innovation. These guys did it. They didn't really have any great ideas about what they would do with it. And I asked the engineers there in 1991 or 2 or something like that, could I use this idea of instrument control language to do automated tandem mass spectrometry? And the engineer I spoke to said, no, that won't work. So I didn't do it. But this guy, Terry Lee, did it. And what he did is he automated the process of collecting tandem mass spectra. And that's something that we do very routinely these days. And I was, of course, watching what Terry was doing and saw that he did it. The lesson I learned from that is when somebody tells you can't do something, they're probably wrong. So we did a different experiment, automated, using an instrument control language where we collected connected neutral loss scans on a triple particle mass spectrometer to tandem mass spectral scans, so we could automatically collect spectra of phosphodipeptides. This was coupled together with search for showing that we could search databases and identify modified peptides using sequences in the protein sequence database. Okay, the other important piece of development was this idea of capillary liquid chromatography columns. This was developed by Jim Jorgensen's laboratory, but also Milos Novotny's laboratory was involved in doing this at the time. And what they were trying to do was to understand the fundamentals of chromatography. There's this thing called the wall effect, where the beads in a regularly packed big column don't line up correctly along the wall, and so you get these defects in the separation. They were trying to understand that by using these very small columns. And it wasn't that they were trying to invent something for mass spectrometry, given the time frame here, but it turned out that this technology that they developed to do this dovetailed beautifully with what had happened in the field of mass spectrometry. So now we could use these capillary liquid chromatography columns to do these great separations. The other thing that I think was important was this idea of two-dimensional separations, and J. Calvin Giddings, I believe, gets credit for having at least inspired this area, if not I worked in it first, this idea that you can do a multi-dimensional separation as a way to increase the separation power. This is, again, another paper from Jim Jorgensen's laboratory where he's got 2 columns put together, and he's doing effectively doing a two-dimensional separation, except he's using a UV detector on this. This was done about 1990 or so. So I'm very interested in this, and we bought this device developed by Fred Rainier. It's a multidimensional separation instrument for doing chromatography. It's got all these 10-port valves, which made it very, very powerful. And we did tried to do a total yeast proteome, and out of that we got 189 yeast proteins identified by the mass spectrometer. This is, again, using an LCQ mass spectrometer, so an old ion trap mass spectrometer. This may not look like a big number, but in 1999, this was a new world record. A previous record was held by Matthias Mann, 160 proteins pulled off of a 2D gel and then analyzed by MALDI time of flight. It took them many, many, many months to do their experiments. It took us 24 hours to get this number. So a big improvement. But clearly, it wasn't very good. And so we thought that the flow rates were not suitable for the process, and that we could do better by reinventing how we do multidimensional liquid chromatography. So we developed this device as a way to do multidimensional chromatography, repeated the experiment, we get an order of magnitude more proteins being identified. That was on the slide that Alexei showed earlier this morning. And so this is 2001 that this is published, and this, again, sets a new record for number of proteins identified. But now we know that we can do this. We can do better than that in 1 hour on the new mass spectrometry technology, the Thermo Fisher fusion technology. Okay, so fast forward about a decade or so, and now we're able to take this same technology and start asking questions about diseases that people are interested in, such as depression. So this is a study that we recently completed with Helen Hu, who's now at we started this when she was at UCSD. She's now back in China at the Institute for Neurobiology. And there are a number of different treatments for depression, and from a lot of cases of depression can be very effectively treated with SSRIs, but there are a set of people that don't respond very well to these treatments, and they have very severe depression, and other strategies people are looking at, such as deep brain stimulation of of the habenula complex. And so it is known that the habenula complex does have a role in both emotions, but also in depression. People have found that this is an area of hyperactivity in depression in animal models of disease. These are electrophysiology experiments shown here, showing the activity of receptors in the brain on this congenital learned helplessness model, which is a model for depression. You can see very high levels of activity versus the wild type animal. Also from these functional MRI studies as well. And so we worked with Hallam to use this congenital learned helplessness model. This is just phenotyping the animals, showing that the wild type animal, this is pressing levers on an electric cage. The depressed animals don't press the lever as much because they have these very low levels of anxiety. They don't get upset about anything, and so they just sort of they're helpless. So they just kind of sit there. When you treat them with an SSRI, they respond just like the wild type animals. So these numbers in the bottom are the numbers of animals being analyzed. And there's also a forced swim model too as a way to verify these phenotypes. So the question we want to ask is what are the molecular changes that are occurring in the habenula complex that might be contributing to the depression? And so we have this way of doing stabilized tope labeling of animals. We use N15. This allows us to do quantitative analysis of molecules, of proteins within these different studies. And so this is the study design. We're going to take a habenula complex from N14 brain, mix in some N15 brain, and then we're going to take from a depressed animal, N14 brain, again mix in N15. So we'll do ratio of ratios measurement, and that way we can control for systematic errors that get introduced in this process, and then we'll get the ratios out at the end, and we'll be able to see what's changing. This is a volcano plot showing statistical significance versus fold change of proteins. There are a few things that are interesting up in here, and I'm just going to tell you one slide about beta-KMK2, which was a catalytic molecule that was found to be changing. So we have lots of catalytic molecules and ion channels and so forth that look like they're being upregulated. We have a bunch of proteins that are being downregulated. It's this KMK2 family that we think is interesting. Without showing you too much data about this, if you look at the habenula complex versus tubulin, you see that it's very highly upregulated. When we treat an SSRI, we can show that the levels of beta-KMK2 go down back to normal. And so this beta-KMK2 was clearly shown to be through a variety of other experiments that were done. It was very clearly shown to be involved in depression. But is this a new target for the treatment of depression? Probably not, because this is a rather ubiquitous kinase that's involved in a lot of different processes, although its target is known to be this GLUR1 receptor, and so that may actually make a better target. So mass spectrometry proteomic data is complicated, so let's refer to some of this. So peptide sequences are used as surrogates for proteins. We have isoforms that need to be sorted out. We have identities and quantities are present in the data. Post-translational modifications are located in there. We also often have secondary information in our data sets, such as protein-protein interactions or localization or activity. And so we need to be able to sort all that out. And so proteomic data analysis pipeline, when workflows have been developed, this is one that we use. So we take the raw files, we identify things, we do ID compares, we have quantitative analysis, we have a variety of different ways we can pull that information out. And then after we've done quantitative analysis, we need to do statistical comparisons. And then the real bottleneck for us has always been, how do we figure out what are the important things to now follow up in biologically? And so that's the functional analysis end of the whole analysis part of it. And so we've been developing tools to do this. So here's some of the tools that we use for computational analysis of the data. The functional analysis techniques, we have a variation on gene sequence enrichment analysis. We have ways to look at gene ontology, David, which is a program out of NIH, and also ingenuity pathway analysis. And then we have ways developing data storage to store all that information. So we have methods to do, as I showed you earlier, we have methods for doing statistical analysis for comparing multiple samples. We use volcano plots quite routinely to show p-values versus fold changes, things which are going up, things which are going down. That can tell us what things to focus on experimentally. We do gene ontology analysis to look at biological processes that might be enriched in our experiments. And then the gene ontology analysis can tell us what proteins are being significantly changed as a way to find things to follow up on. So the limitations of gene ontology analysis: it assumes gene and protein expression independence, does not consider gene protein expression or abundance, requires establishment of arbitrarily chosen thresholds, and often yields long lists of enriched Go terms. And Go terms are not a very highly resolved term for proteins, and it's rather a broad swath of information about what proteins might be doing or might be involved in. And so Matthew took this gene set enrichment analysis and adopted it for proteins, but also using quantitative information. And this gives us a way to look more precisely at what proteins are doing as a function of their quantitative changes. This gives us a little better, more highly resolving analysis of the proteins that are changing. So this can be applied on samples originated from a single or more conditions. Does not require arbitrary thresholds. Models the likelihood of a protein being annotated by a certain protein set, and models protein abundance correlations much better. So we also have a way. So one of the problems that we've had in the lab for a long time is that when somebody does one of these great big huge studies and they leave, the sort of institutional memory associated with those projects leaves with them. And so what we've been trying to do is to, rather than have the data stuck in Excel spreadsheets and things like that, we want to develop some sort of long-term storage of the data so the data doesn't die when the scientist leaves. This would also provide us a way to share out this highly evolved or manipulated set of data out to the scientific community. Like we recently have a paper that we're trying to get published, and one of the reviewers came back with a comment of, "This is more data than I've ever seen in a paper before in my life. How are you guys going to provide this to people?" And so having a way to do that also has been something we've strived for. So this will also enable higher level analysis of the data, so we'd be able to compare, say, a depression study on brain in rats with a schizophrenia study that we might be doing on brains in rats too. So we'd have that be able to ask questions between data sets within the laboratory, where we've got a little more control over how the questions are being asked. This will be a web-based tool for storing and sharing final proteomics data, be a local repository. We're having a flexible data model so we can do all kinds of different types of data, from differential expression data, cross-link data, protein interactomes, and so forth. Also things like phosphoproteomics as well. So we'll be capable to import proteomic results after manual or non-standard user interventions. In other words, our use of in-house tools and so forth. This will be a tool to manage and share the huge amount of data generated by our proteomics laboratory, and we should have high-level analysis of data sets supported by a sophisticated query system. In part, when we publish our paper, we want to be able to provide biologists, so people who are not going to be interested in taking our data and reprocessing it, to have them be able to come in and start asking specific questions about our data sets that they might be interested in. Does my protein interact within your data set, for example? Okay, and then these are people in the laboratory that have been doing the work. Matthew is there. He's also there. And then Robin is, where's Robin? Taking the picture? Oh, there he is. There's Robin. And then, let's see, there's Lynn. Lynn also works on software. And let's see, who else? Oh, Salva. Salva's been doing the PINT and the protein integrator. So thanks.
So John, for your this PINT, what does that mean? Like, so we're doing something similar, but we're just putting it in an Access database right now, and then going from there. So is this a software or?
Yeah, it will be an SQL-based software
SQL.
tool.
Okay.
So it would be like Access, except it's the higher, little higher end.
Yeah, because we're trying to merge not just proteomics, but also we have a lot of biology, a lot of physiology. And so we're trying to get 1,600 mice worth of data into a system where we could do something similar. So we can share those.
Yeah, the biggest problem that we have is the institutional memory associated with these projects.
Even when the person doesn't leave.
Yes. And I recently had somebody leave the lab, and she didn't write all the papers, and we're trying to get her to write the papers. The data is all on Excel spreadsheets, on her computer.
Yeah. So John, I see this then fitting into sort of the protein exchange environment. Time specific.
Oh, not at all.
Not at all.
No. And it's not intended for that. These guys are doing a great job. These guys are doing what they're supposed to be doing. This, for us, is more of a in-house tool where we can deal with our data sets and not lose them to tape or, hard drives like that. And then share, then when we publish the paper, we can share out those data sets which have been highly analyzed and curated, manipulated, whatever. So share it to the paper. The data behind all that would go to proteome exchange separately.
So this is prior to sort of the time at which a lab would want to distribute that data or share it, but it's more like within a given lab, the data management engine. Is that right?
Yes, and this would also be able to support a paper. So the paper gets published, people will be able to ask additional questions. Because oftentimes we've only dealt with a couple or a few proteins in terms of functional validation. And so we need many more things which should be validated. People want to ask more questions about that. So this is intended to be a repository like proteome exchange. It's intended to support the efforts that are going on in terms of understanding the biology.
So that to develop this, is it meant to be for public access, or is it meant to be only local and private as part of the publication process? It will go into the
Yeah, so once the paper is published, that part of the data will be available publicly from there to support the paper. But the raw data will also be submitted to repositories.
I understand you want to keep the data files, but what about the tools and the methods that you develop? Maybe interesting to share, to teach to others, or vice versa, to learn from others about methods and tools.
They're all available.
Okay.
So your approach is yours now for protein segmentation analysis. So how's it different? I guess what do you gain compared to just mapping to gene names using the standard tools developed for gene expression?
You didn't read our paper?
I did, but still, I want a 15-second summary of that.
Exactly, 10-second summary.
One of the main things we find is that we take advantage of the proteomics, the proteomics abundance values, and we model the variation of these values better than if we were to translate it into gene name and use GSD, for instance.
But there are also tools that take the balance.
Right, so PSC, the first generation PSCA, for instance, but also that we found in our lab, we don't have data sets that are just characterizing certain organism or certain, it's just certain organism. We're not comparing two different conditions as an exactly PSC and GSD strategy. So we develop this spin-off basically that helps you to only see an enrichment when you're not doing comparisons of two conditions, for instance, which is something we often do in our lab and the audits lab.
We try to get the published PSC data set. So the Aaron Carter's lab published this sort of program version of GSDA. Couldn't get the guy.
Yeah, we never got the code for PSCA, original PSC function, original. So we had also a service that PSC for everyone.
Well, the one thing that we found makes huge difference is where he uses the right chromosomal as an alpha straight. So this is a variation instruction. He uses the whole genome because the hemolytic process profiling is pretty much everything. This proteome is so dependent on differential expression in the data set. The proteome
I'm sure you yes, you can use the background is basically your data set. And you don't use the entire database in the background.
So there are a variety of we previously collected this intelligence tools, but now they're called data science platforms. And we would back with that last slide suggestion to just put all the data into schema format, and you have an analytics layer that they so things like business intelligence tools or data science platforms. Yeah, so like a hedge fund for a financial institution, they have a variety of data sets that they look at. And it's pretty common for them to fuse them all together. So there's this company in the Bay Area called Palantir, who the NSA uses and Big Oil uses and a variety of other people who have a variety of data sets to, again, store the data and then have a very useful user interface for the analytics layer. And there's a bunch of startups in San Francisco and Boston and Canada that do this. So if you look at any of those, well, so the smaller startups usually have an accident rate. And then Saccomani's ITV2, like for the physiology data, which is essentially just an electronic record that's coupled.
That hasn't been the rate limiting issue for us. The rate limiting issue for us has been making sure we understand the data before it goes in and cleaning the data, making sure it's as clean as possible. And so, for example, if we measure mRNA levels, did we do it by hydrogen array, or did we do it by PCR, or did we do it by, so those kinds of things, you have to think how the data goes in, because you have to organize, if we want to mix and match and cross everything, we need to make sure that it's really mixed and matchable. So those kind of things are what's been our rate limiting step, not just putting data in something.
Well, that, so like Miami standard for microarrays is very useful, right? Because you have all this metadata about the experiment, but that needs to be done when the experiment is finished.
Yeah, it is. It is. But just because we change platforms, we change, companies, we change stuff like that. So we just need to think how it goes in.
Metadata is, to me, is the absolute key to putting on a data set because people ask them much more sophisticated questions, detailed questions. And so if you don't have that information, the data set doesn't mean much.
Are there any further questions for Dr. Yates? Okay. Thank you, Dr. Yates. So that does conclude the morning portion of our current meeting. We are going to proceed and have lunch outside on the courtyard. A couple of you went up inside, but we're going to have lunch from 12:30 to 1:30 p.m. And there are 2 working lunch groups that need to be looked at. There's one specifically for the education component, one for the DSR component, but people are welcome to eat with, join in on the meeting if they feel like they can contribute and want to listen and all things like that. We have sandwiches over there. There's turkey, tuna, roast beef, and I think a vegetarian option, as well as sodas and waters. So thank you very much, guys.
We got started up on All right, so welcome back to the afternoon session. So this is going to be part 2 of the data science tool session. We're going to have 3 talks followed by a panel discussion on clinical cohorts. And the first speaker this afternoon is Dr. Mary Lindsay from the University of Mississippi, and she's going to talk about the Jackson-Hart study and omics analysis. Thank you.
And I was getting this talk ready, and I only have 13 slides. I was thinking just to go on to the Jackson-Hart Study website and show you stuff. And that's kind of what I'm going to do, but I went ahead and put it as a PowerPoint. And so I want to basically show you guys where you can find the information. Okay, how do I put it?
There's a clicker.
Okay, I can do that. Okay. Okay, so the Jackson-Hart study, someone just said to me, what is that, like the Birmingham horse study? Well, yeah, that's what it's like. It's a large community-based observational study of urban and rural areas of the surrounding Jackson, Mississippi. So there's 3 counties, Hinds County, Madison County, and Rankin County, that were included in the study. And enrollment occurred from 4 different pools, which I'll show on the next slide. And so non-institutionalized adult African Americans 21 to 84 years old were recruited. So everyone in the study is African American. The final cohort is 5,301 participants, and this represents 6.59% of all African Americans in Jackson. So the enrollment was through 4 different mechanisms. One was a random mechanism where they took the acute AccuData American commercial listing and basically tried to find people to recruit into the study. They also accepted volunteers. So Herman Taylor was the PI of the Jackson-Hart study up until about 2 years ago. He's now at Morehouse. He did a very good job to go out to the community and tell folks why it was important to be involved in this study. And then also there was a study that was funded by an HLBI, the ERIC study, atherosclerosis risks in communities, and 30% of the Jackson-Hart cohort are actually from the ERIC study, so they just continued since they were already enrolled. And then as a second phase of the Jackson-Hart study, there was a secondary family member enrollment. And so the initial phase was, I think it was 34 to 80 whatever year old. And the second one, it was, went down to 21 years old. But the secondary family members all had to have a family member who was enrolled in the original cohort. So the major exam components that were taken, there's 3 visits to date, and at each visit there was a medical history taken, a physical examination, and including starting at visit 2, it included echocardiograms. Also there was blood and urine analytes measured, and then interview questions asked. Like I said, 3 visits have been done to date, and then also in between the visits there was every 12 months there was phone interviews. And then there's also ongoing cohort surveillance, and I'll tell what that means too. So at the visit interview, questions were asked about the physical activity, the stress, coping and spirituality, racism and discrimination, socioeconomic position, and access to health care. And so those were just verbally asked, verbally answered, and has all the plus and minuses that go with that kind of mechanism. During the annual phone interviews, participants were asked to update any information and to confirm their vital statistics, and then tell the interviewer if anything had happened to them in terms of medical events, hospitalizations, and functional status. and then also to obtain additional sociocultural information. So again, phone interviews are as good as the answers. And I know on the driver's license, the body weight, for example, and height might be good examples of what you say is what gets put on the form. And so we also have mechanisms for ongoing cohort surveillance, which includes using medical records and death certificates and classifying by classification of disease. So for example, heart failure is now adjudicated heart failure is one listing that gets put into the Jackson Heart Study. So in one case, someone could say, oh yeah, a doctor one time told me I had heart failure. But also, if it's adjudicated, that's also put into the record. And if we're going to use heart failure samples, we want the adjudicated ones as our records. So this is the website for the Jackson Heart Study. You'll notice that it's not a umc.edu website. I was talking this morning about university websites, and it's definitely true at UMC that it's easier to set up your own website or have your own Facebook or whatever than it is to go through the bureaucratic procedure. And so if you go to the umc.edu homepage and you look for the Jackson Heart Study, you'll have difficulty. So what you got to do is just, if you Google Jackson Heart Study, this is the website that comes up, and you'll get a page like this. And the for researchers is what you want to click on. And so that for researchers is the key link that tells you where information is at. And so in terms of navigating the Jackson Heart Study, here's some listings of what is available. The publications, everything that's been published using Jackson Heart Study data, you can get a list of it there. Ancillary studies, this is more how, and I'm going to actually show you. Ancillary studies, how to have an ancillary study protocol approved. And so KK and I started last April to do the paperwork. So on the website it says it takes 3 months, but in between there's time of doing stuff that's not included. And so straight start from finish with no gap in between, sure. But you got to realistically give yourself a year to go through some of the hurdles. Also data access, data summaries, those are all stuff that you can find on this website. It's just an overview of the ancillary study proposal procedure. You don't need to worry about that too much. Because like I said, for the big data contract, we already have one in place. It's just a matter of adding people if we need to, or and we're IRB approved to do some proteomic analysis of some of the Jackson Heart Study samples, but also to get access to the data sets. And so this is just showing what the data summaries look like. And so you can just go on and anybody can look and see for each of the three data, each of the 3 visits, what the cumulative data look like. So it's not the individual data, but cumulative is posted on the website. And then you can also look at study components here. It tells you for each of the 3 exams, the first exam was in 2004, and then the second was 2005 to 8, and then the third was 2009 to 12. They had standing height measured each time, and it goes down the table what the different measurements were. So all of this data is available as de-identified results. And so it was expedited IRB review that took us 3 months, but that's another story. Anyway, we have access to all of this data. And in our ancillary study protocol and in the IRB, we said any information that Jackson Heart Study has available, we want. But here's an example of what we're going to want. So we left it broad enough. We were talking this morning about when you write your IRB protocol and you write your proposals, always say, I'm going to measure X, Y, and Z and something similar or anything else that I can. Anytime you can have wording like that, it really helps for people to give you access. Another resource that's interesting is the Candidate Gene Association resource here. And in the CARE one, there's been Jackson Heart Study samples that have been evaluated. So this is a genotype and phenotype database that has 41,000 study participants that measured about 50,000 SNPs from over 2,000 candidate genes. It's also done GWAS for about 8,900 subjects, including, and they're all African Americans from these 5 cohorts. So included in the Jackson Heart Study. And actually, Jim Wilson, who is a member of the Jackson Heart Study, was just telling me that they've been approved, they've been funded to go ahead and do GWAS on every participant in the Jackson Heart Study. So that's data that will be coming along eventually. And so this data from individual cohorts is available to approved investigators through this kind of mechanism. And so 2 people that you really need to know for Jackson Heart Study, besides me, Mike Griswold is the director of the Coordinating Center, and he's also director of the UMC Center for Biostatistics. And so he's the one who has his fingers on the data. And so we're talking about how to, get access to those data sets and start playing with them. He's our buddy. He's the one who, he has a group of statisticians who can actually give us those data sets. And then Jim Wilson, who I mentioned before, he is heavily involved in the genetic analysis, and so all the GWAS studies, et cetera, he's the contact for that. And so that's my little summary of Jackson Heart Study. It's always good to know how to find information, and I hope that that's helpful. Thank you.
All right, so the next speaker is Chunlei Wu from Scripps Institute, and he's going to talk about my gene info and the wildly cohorts. Thanks.
Thanks for the organizer of this meeting, and it's very honored to join this BD2K project. I'm looking forward to working with all of you guys. And yeah, I'm going to talk about the first. I'm going to start with the wildly cohort. Great. So yeah, I will start with the wildly cohort. That's actually done from the Scripps Translational Institute. I will be presenting this on PowerPoint. And also, then I will continue with 2 tools we developed in our group and will help to distribute and query for the large data like wildly cohort and all the other data be generated in this class. So, wildly study actually led by Dr. Eric Topol from Scripps Translational Science Institute. Actually, if I remember correctly, it started from early as 2008. This study to basically is one to recruit people who's healthy and live longer than the general population. And there's the criteria that we recruit people that know chronic disease like cancer, diabetes, and all these issues. And currently the cohort is about 1,600 individuals who report 50 clinical phenotypes. We also use genome sequencing on all these people, and with first actually done more than 50 patients with a complete genomics platform, and we've basically done the 200 people on the Illumina platform. There's the data available to download, and there's also genotype data. There's also genotype data and also cell line data, cell line available for the site. And this is a summary of all this characteristic for this wildly cohort difference between the wildly cohort and the general population. It's actually very interesting to see. It's not surprising to see exercise and the education will play a role in this wildly cohort. But it's interesting to see this smoker, higher percentage of smoker in the wildly cohort compared to the general U.S. population. So,
smoking is not like
that's something from the school. I'm not sure if this is a conclusion. Yeah. And also, this is another data shows the elderly siblings survival compared to the general population. You can see significant better survival rate for the elderly siblings. That's also not surprising. And then this slide just show like for the point of view like genetics or genomics, we want to look for in this cohort actually lack of a disease causing variants, and or if this cohort actually contains more protective variants. That way we can hopefully solve the people like aging issue. The initial study right now we show that actually there's no evidence of lack of disease variants in most people, except the E4 variant variants that show up lower in the in elderly cohort. And right now we also have the pending study for looking for the protective variants in this cohort. This is also some challenge based on what type of data set will be the reasonable, the suitable control data set. We are working with Innova companies to have this control data set. Yeah. So it will be very interesting to use this elderly cohort in this BD2K project to see if there's any overlapping with other type of genomics data set. And right now, elderly variant data, we already collect onto all this variant data. Now we have about 21 million variants. And we have a simple query interface right now in the elderly toolab.org. You can query by the position, query by the gene, very basic, and putting more annotation to this interface in the future. Also, for the documentation, we also provide programmatical access using the myvariant.info platform, which I'm going to talk about more in the next few slides. For example, you can use all this syntax, REST-based web service to access all the elderly variants information. And with myvariant.info, you can also query between the elderly variants and overlapping with other existing database variants and database. For example, if there's any overlapping variants with elderly and BD2K for the missense mutations. Yeah. And since I already mentioned myvariant.info, I'm going to talk about this myvariant.info infrastructure we built. And this is essentially this is a high-performance query engine for aggregated variant applications. This is the website, easy to remember. Because we know all the variants right now is all over the place, and bioinformaticians spend a lot of time actually aggregating data together, and actually they are doing this repeated effort. It's very time-consuming, much more time spent on the same repetitive work than doing the actual analysis. So we have the same similar situation. That's why we come up with a platform like this, trying to help us and also help the research community. Basically, in myvariant.info, we try to we basically model all the data, variant application data in this so-called JSON. This so-called document in a NoSQL database system, or we can a variance annotation object. The ID will be something like this: the chromosome 1 at this position, C2T mutation for this one. And the DB-SNP is just one source of annotation that we list all the information for the DB-SNP. Now you can imagine we can aggregate all the information from other databases, COSMIT, DB-SNP, DB-MSP, all this information, and together with the elderly, so that when we loaded the elderly data, we know all this match information from all these existing data sources. And this particular items. This slide will show you the example of elderly data. Right now, we we recorded it in our myVariant.info database. The well-data data, we have all this basic information. We don't really need to load all the other information, other annotation information like polyphen annotation score or the symptom score. Because we already have this in the database, there's a match in the database, we got all this together automatically. And as you already saw from the last slides, we use the so-called HGS, Human Genome Variant Nomenclature, as the ID to aggregating all the variant annotation. This is a slide just to show you some summarizing the common variants type we can present the data using the HGS ID. Okay, and this is just giving you a sense of the current variants we loaded in the variant platform. We have more than 116 million right now loaded, and including the worldly. We also have a bunch of new sources in the pipeline to load. And before I talk about a little more like back end of the myVariant.info, so you mentioned my Gene.info now. So now we have myVariant.info for the variant annotation, and Gene.info is actually for the gene annotation. The myVariant.info actually built on top of the improve the infrastructure of the Gene.info, which I highlighted basically in this box. It's like a JSON representation of heterogeneous data, an up-to-date data aggregation platform, and plus the high-performance data query engine we put on top of the aggregated data, so that we can provide a very high-efficient query interface for most of this service. This is my Gene.info page. We keep the data very up to date, and this is I actually just repeat what I just mentioned for the myVariant.info. We want to isolate this data wrangling step from this task from the bioinformatician, so that we don't really need to have to repeat all this repetitive work. And we want to isolate this as a web service. Now, to make the web service really suitable for the real-time applications, we want to make it fast enough, and it's always stable, there's always data is up to date. And so we, just like we did in the myVariant.info, we use this structure to represent the gene annotation, and this is a more like a real example for the gene annotation. We aggregate a bunch of this. You can see every item here actually is already collapsed, so you can see the whole information about this same gene all aggregated in this same document. And now we have a data hub component that will take all the information from the issue resources and do the parsing and have the individual annotation from the resources, and then have to do the merging into our query instance and serve to the end user. And in reality we need to build a cluster of the query instance so that we can achieve a really high performance. And this is my Gene.info API for the developers can use in their application. I won't get into the details of this. And this shows my Gene.info data coverage. Right now we support all species with the gene annotation. That's like more than 50,000 species for 18 million genes. And we loaded more than 50 annotation fields that's also expanding. And as the data is weekly updated, we always keep this up to date, and also that data always accessible. The flexible query interface is close to the end user. All the common query different type of query syntax can fit into the different use cases. And more details can be found from my Gene.info documentation. And this also shows a very high performance host that we offer for the Gene.info. Right now we have serving about 100K requests a day. This is we have a really high concurrent user record in our website. And this was just to show the stress test that we've done for the Gene.info. We can support a 5,000 concurrent user for the 10,000 K requests per minute. This is like averaging like a request per month on the real use case. This one I want to mention, like actually during just the last Christmas, we have someone really working hard to take our web service. 5 million per day. That's almost like doing our stress test from our direct user. We're really glad that our server really coded up for that and it's still working okay. Yeah.
Trace down all the IP address.
Yeah. So probably like not one particular, it's like one or two of this. I know that one of this I can know is like from Korea. So yeah, they're probably don't know. Server is like Christmas. Yeah. Okay. So query, this is a real world query performance. Like most of the query performance that we done for the real world query performance is done within the 10, 20 milliseconds. It's really fast. I won't mention too much about this, but we have the Python package, we have R package, and also have the JavaScript widget, so that we can a lot of also other applications in our group and build on top of the mygene.info or myvariant.info APIs. And also last I want to mention the mygene.info as a platform, also the myvariant.info. What does this mean for the data provider? Actually, data provider for for people who generate data, you can provide the data to your specific data to the platform, so that you can achieve. You don't really worry about all this IT infrastructure and how to build the sophisticated software to develop make the platform query interface and high performance. And also you can have the benefit of the automatic aggregating with all other annotations. This is one example of data importer contributed from the communities. It's really simple. Also, I want to mention this platform is neutral to all the other different types of biological entities. We can easily get extensive like gene variants we have already done. We can also start thinking about disease or metabolite, even maybe like patient information, maybe. And also want to reference what Andrew mentioned about the network of biases. Because all these biological entities you are linked to, all this relationship is also very important. So we want to also incorporate semantic web technology so that all these components are linked together and just become the one solid component in this network biases structure. And I would like to thank all the colleagues from Andrew's group, and also our developer, and also the SDSI operator, and the from the Thanks.
Thanks, Shunli. So our next speaker is Jessica Zhu. Chelsea Zhu, sorry about that. And she's from Wei Wang's lab at UCLA Department of Computer Science, and she's going to talk about data analysis for heart failure patients.
Thank you. Thank you everyone for coming. It's a great pleasure to present our work as part of the BD2K project. And I'll present the work which is entitled Automatic Data Analysis of Heart Failure Patients. So to give you a little bit of background of this project, there are more than 6.6 million of heart failure patients in the United States, and 80% of them are hospitalized, and 50% of them are actually 50% of them However, many patients develop multiple organ dysfunction syndrome after the implantation, which is actually the major cause of that for this treatment. And the advanced heart failure is often clinically treated as a pathological disease with similar treatment options. However, recent studies have suggested that the heart failure disease is actually heterogeneous syndrome that might require divergent therapies. An accurate phenotyping and classification of the advanced heart failure is still preliminary due to the lack of quantifiable and validated methods. And some of the metabolism, this slide is basically showing the relevance of why we use the metabolism study. It's relevant because it can serve as a guide to biologists as well as discovery related back to diagnosis and therapy. And we can also use it for patient stratification, and clinical classification. And it can also provide information for mechanical insight of complex pathophysiological, cardio, and remodeling studies. And there are many other more advantages to using a metabolism study. And being that the end of the study is trying to use the metabolic profiles to identify and characterize the factors that correlated and contribute to different clinical performance patients after the implantation. So this slide shows a brief summary for the data that we see. The data comes from a time series blood sample collected from healthy participants and 26 advanced heart failure patients according to New York Heart Association classification. So in other words, they are in the worst stage of the heart failure. And 611 metabolisms are quantified and screened for every sample. And the pie chart here basically shows the demographic information for our individuals. And there's 77% of the patients who are male, and 23% of them are female. And among our healthy participants, 67% of them are male, and 33% of them are female. In terms of the rate, we have the rate of our individual participation, African American, Asian, and Hispanic. And as I mentioned, this is a time series study. So the time point, so the individuals are sampled at different time points. One of the time points before the surgery and also after the surgery, up to 120 days. So here's the chart to show the time interval between the time points. So we use T1 to denote the sampling time before the surgery, and T2 corresponding to up to 5 days after surgery, and T3 corresponding to up to 11 days after the surgery. And most of the healthy participants are sampled up to T5, but patients are sampled as long as they are hospitalized. Some of the patients are performing really well, so they got really good, and some of them didn't make it to the study, so we don't have a country sample for those patients. And in terms of the data analysis, in the process for developing the methodology, we only use a subset of data. And this subset of data contains all healthy participants and 15 heart failure patients. And we only focus on the time point from T1 to T5. And the analysis includes expression analysis, pairwise correlation analysis, and principal component analysis. We can now go through each one of them on the next slide. So when we receive the data, one of the questions we want to find is where we want to see whether there's a differences between the healthy participants in terms of metabolic profiling between the healthy participants and the patient before the surgery. So we use differential expression analysis with the goal to identify a set of statistical significant differentially expressed metabolites between the healthy individuals and heart failure patients. And the approach we use is to apply a frequency test on each metabolite to evaluate the significance of quantification changes between the healthy participants and our patients. And since we apply a statistic, the statistical test on every single metabolite, we have to correct the multiple correction problems, and we use Benjamin Hopper procedure to control the active variation. And after all that, we select the final list of metabolites using two parameters. The first one is the p-value after the multiple test with correction, so the p-value is less than 0.05. And another parameter we use is the absolute protein. So if the protein is bigger than 2, we pick those metabolites. So using this approach, applying this approach on our data for the time point before surgery on T1, we identified 196 significantly differentially expressed metabolites, demonstrating this volcano plot here. So the x-axis of this volcano plot indicates the total change between patient and healthy participant in the log 2 scale. And the y-axis shows the p-value in the negative log 10 scale. The two vertical lines that we see on the graph indicating the cutoff for total change, and the horizontal line in the graph shows the cutoff for p-value. And we see 80 top record, so 80 on the top right are top regulated in the patient, as indicated by the blue color in this plot, and 116 metabolites are downregulated in the patient, indicated by the red in this plot. And the gray dots over here basically mean they are insignificant, so they are not picked up on a specific test. And the next thing we want to do, I'm trying to find out is we're trying to explore the expression pattern for all the differentially expressed metabolites we identified. So we use a heat map and a hierarchical cluster to demonstrate this. So each row the rows represent the metabolite, and the columns represent the individuals. And we can see that the hierarchical clustering actually groups all the individuals, all the patients together, as indicated by the box over here, down and it also groups all the healthy participants together, as indicated by the blue box. And we also see that in the metabolites, all the metabolites with similar expression patterns are grouped together. And it also shows that the metabolite shows a very distinct pattern between the patient and the healthy participant. And next, we try to figure out what those metabolites are responding to, like which classes they belong to. So we plotted everything the bar over here. So on the x-axis shows the different classes of metabolites, and the y-axis shows the boundaries of each class. We see that the differentially expressed metabolites are identified across 20 different classes, and a large number of phosphatic choline are downregulated in the patient, as indicated by the red arrow over there. And a high portion of plastic kinase are upregulated in the patient, as indicated by the green arrows over there. So phosphatidylcholine is actually by far the most actually sources of choline in human. The actual mechanism related to heart disease is still unclear, but choline is linked to the synthesis of homocysteine, which is one of the known risk factors for cardiovascular disease. And the deficiency of choline in the thiamine, may lead to lipid accumulation in liver, in heart, and also in arterial tissues. And there are some studies suggesting that if we're manipulating the metabolism of phosphatidylcholine may be enough therapeutic approach for heart disease. And also another research that suggests that one of the phosphatidylcholine species, the LPT, is a promising therapeutic agent to improve diabetes. And for antiochronitin, it's a class of essential compounds for fatty acid metabolism. The direct link to heart disease is still unclear, but it is known to be involved in fatty acid oxidation. So moving on our next analysis, which is more like an explorative analysis. What we're trying to do is to hopefully provide correlation to explore the expression patterns for all the metabolites across different time points. So the first thing we want to do is want to retrieve the expression pattern for each metabolite. And here we define the profile for each metabolite by the expression and by the quantification across different time points for different individuals. So here's the table to demonstrate how we define the profile. So each row represents each metabolite, and each column represents a specific patient at a specific time point, and each cell is the quantification of the metabolite corresponding to the metabolite and the patient. So we define the expression profile of the entire row. So for example, for N1, the entire row of the expression level of the entire row, we define that as the expression profile for N1. So taking all the expression profile for all metabolites, it's confused that there's some correlation between every possible pair of metabolites, and the map is put into a correlation matrix. As we can see from the correlation matrix, the diagonal matrix represents the correlation to itself, so it's all one. And the values on the diagonal are symmetric to each other. Then we apply hierarchical clustering on top of this correlation just to reorganize the metabolites. So the metabolites with similar correlation coefficient will be grouped together and displayed next to each other. And use a heat map to demonstrate the results. So the dark blue color here indicates a strong positive correlation between the two metabolites, and the dark red color here shows a very strong negative correlation between the two metabolites. So applying this approach to our data, we see this giant pairwise correlation heat map here. So again, the x-axis and y-axis are the metabolites, and they are ordered in the same order for the x-axis and y-axis. And the diagonal, the dark blue along the diagonal indicating the correlation with itself. And we can also see the dark blue patches along the diagonal, which means that those two metabolites shows a very similar pattern with each other. And from this heat map, we can identify 5 subgroups standing out from the heat map. And we want to take a closer look at those 5 subgroup regions. And some of the subgroups actually are dominated by the metabolites of phosphatidylcholine, as I mentioned earlier, corresponding to the analysis. So we try to plot out the changes of the expression of all the features in this process. The x-axis over here shows the time of the days from the surgery. So 0 means the day of the surgery, and the negative days means the time before the surgery. And the positive means the time after the surgery, the number of days after the surgery. And the y-axis over here shows the expression change relative to T1, which is the day before the surgery. So we just plot out the change a long time. And different patients are depicted by different colors in this graph. And another interesting group that's being identified from the map, which is a small little blue patches along the diagram, which are dominated by the metabolite of metabolism. And again, we try to plot out the differences of the metabolite changes across the different time periods. So, a couple of words that we're working in process for coming to this correlation analysis. I want to further those big subgroups into a small subgroup because they might be some more interesting patterns diving into the subgroups. And also we want to perform an enrichment analysis to do some functional annotation and to examine the functional impact of the process. And another question that we're trying to answer is that we want to be we're interested to find out what are the set of influential metabolites that can be distinguished from the healthy participants. And for this analysis, we use a principal component analysis, trying to fish out with the hope to fish out the half-life that is influential between the two different populations. And the first thing we do with the data is that we have to apply a data normalization because the absolute quantifications from the half-life vary a lot for different half-lives. And the normalization is simply done by subtracting out the mean values for all of the half-life across different time points and the ratios, and then divided by the standard deviation. And after the data normalization, we apply the PCA on the normalized quantification and project the individuals into the new coordinates based on the first two components. Since the first two components present the most variation among the data. And the patients are further divided into three different groups based on their clinical performance, which is judged by a clinician in UCLA. And we label them as poor, medium, and good. And each time point is plotted separately. So we plot the results in a 2D plot. So this is the result for 21, which is before the surgery. And the x-axis shows the first component, and the y-axis shows the second component. So we project the data into the first two major components here. And as we can see, the PCA is able to separate the patient and the healthy participant very well. However, the distinction among patient performance is 0 to 100. And we apply the same analysis to the different time points we have up to T5. And as we can see that for all of them, we can separate out the patient from the healthy participant very well. But still, the separation among the patient still remains. And as I mentioned, this is actually one of the limitations for PCA. We want to enable just using the first two components, we want to be able to separate the patient based on their clinical performance. So one of the potential solutions we have in mind is trying to look beyond the first two components. Maybe the separation or maybe the distinction actually exists on the later component. And another limitation is that we were unable to pinpoint the specific metabolite corresponding to the separation. So we want to look into other explore other techniques for combination techniques with PCA for patient extraction. So moving on to our future work, this is the work that we have in mind in the is that it's focused on differential extraction analysis, specifically on comparing healthy participants with the patient, and also on comparing the metabolic changes between different time points versus the data for the surgery. And also, we would like to incorporate other types of data to augment our finding in the modeling analysis. For example, we have actually an ongoing project on the mouse, so hopefully we can find similar findings in mouse with a human. And we're also thinking about incorporating some genomic data to correct the population structure in our model. And with that, I'd like to thank my supervisor who supervised the data analysis of the project, and to Pete, who is one of the PhD data analysts to help me with the analysis. And this is a very close collaboration with Dr. James Scott. So especially thanks to the campaign and Dr. David Lynn, who helps a lot with providing the clinical information. And our work as a PhD student, who helps a lot with the bioinformatics analysis and stuff. And Dr. David, Dr. Charles Carroll, and Jennifer, who gave me a lot of insight about the experiment and also the metabolite. Thank you.
So let's thank Chelsea and other speakers for this section. So I think now we have the panel discussion led by Dr. Carol Watson. And I don't know if we want other speakers to come to the front as well. So.
Yeah, I think it would be nice if we could get all the speakers to come up to the front.
I think we already have some questions.
Will we go ahead and any questions?
Yes. So regarding the last presentation, have you considered doing your clustering analysis on all 611 metabolites
just to look at the patient information surrounding the
That might have a distinction there in the PC analysis.
Yeah, definitely. Thank you.
So I have a question about the last presentation. So do you have any data where the healthy patient has transitioned into, or a healthy person has transitioned into a patient?
No.
Okay.
I believe not be data.
No, those are two separate groups. So those were healthy volunteers that entered the study, and the patient group are obviously patients that we found in UCLA. So there's no transition. I know what you mean. You would prefer another group, maybe a between healthy and advanced heart failure. Is that what you suggest?
Yeah. Well, so the healthy people are not hospitalized.
The healthy people are not hospitalized,
but the chronic heart people are.
Yeah, those are really in the advanced heart failure. They are hospitalized.
And are they in ICU or are they in
They are in the ICU and they are on the heart transplant list, actually.
Okay. So then those massive differences that you see with the PCA analysis is likely an effect of the hospital, maybe not heart failure.
It is.
Well, it's likely a reflection of how sick they are. So they're hospitalized because they're that sick, and they just had surgery, so there will be some differences from that point as well.
Right. So a good control would be someone who's had surgery but doesn't have heart failure.
True. Or somebody who has heart failure but isn't that sick.
Right.
I think the surgery would probably be such a massive.
Well, you guys are the T1 group, the first group, 5 days, you have massive immune responses and everything. But when you wait it out to 30 days, 31 days, 45 days, you gradually see the differentiation.
So if you go back to the time course plots.
Sure. This one or the time point?
Yeah, well, that one. Yeah, the linear plots that are showing the metabolites over time. Yeah, so the drug.
That's the day of surgery.
Right. And you see
the
big,
right. You see, it's going to be
that's perioperative.
Yeah, but we are actually dividing all the time points into 3 time frames. First, day 0 till day 5. That's actually 5 days after the surgery. Most likely that's a response of inflammation or recovery after surgery. Then we have another time frame that we are planning to analyze is day 5 till 1 month.
Yeah.
And then long term, day 1 month till 3 months. But all the 3 frames should have an effect on recovery, basically. But I agree that, you see those fluctuations, that's part. That's basically based on recovery after surgery.
Great. Thanks for those excellent presentations, guys. So now let's just have a discussion on some of the unique issues about dealing with clinical cohorts. You've had, I think, some interesting ones came out of your presentation. One of those is it's very difficult when you're dealing with patients to find a really suitable comparator group, right? Have you guys had any discussions about other groups to look at compared to the advanced heart failure patients?
I'm not quite sure, but I know that we have actually had a lot of experience. Hopefully that we can find that being a little bit more considerate because with participation it's really hard to find a real control because getting back to healthy participants for consideration is healthy. So that's how you want that issue.
Okay, great.
So, Carol, that's exactly, so we've designed a set of mouse model experiments with Dr. Lim and Dr. Imam and Travis. What we've done is using that as sort of a parallel analysis and doing temporal profiles where we have more room to manipulate the time points or the number of days post-surgery. Also, that's when we collaborate with Jay Bruce's group, adding in the genetic contributions, looking at different genetic backgrounds and see how genetic variants may contribute to modification of the phenotypic outcomes. So right now it's a sort of closed translational study design with mouse model and with your cardiovascular, all that cohorts.
actually been used.
If I remember correctly, you said in one of the last phrases you are hoping to include genomics data to correct for the population structure. Can you elaborate?
That's an idea that we just came up with. We haven't really spent that much time in terms of how models being built, but we can see that the demographic information from the patient and the health participant. So we see a wide ranges of races that we collected from the patients. So they are Hispanic, Caucasian, Asian, and African American, and others. And seven of healthy participants. But sometimes that the variation can actually be due to genomic differences. So we were actually thinking about incorporating the genomic differences to correct the population structure in terms of to kind of remove that that variation, just to look at directly at the responses, how long responses directly to the failure of the implementation, and not based on
I was going to suggest that the heart donors would be the group that had a healthy heart, and you could still prolong their life after the heart's been transplanted with a ventilator.
Yeah, we should talk about that because we actually do get blood from all of these donors. So. But it's better than many of them just have nothing or something.
Or drug overdose, which would be less.
So it says for the healthy participants, 75% are other.
Is it other or Asian? I can't decide.
Asian.
It's Asian.
Oh, I was like, wow. Sorry. Okay. I was like, what?
I think it's because some of it is us. You probably know that's why it's such a huge Asian.
That was my guess. I didn't want to ask.
Population and well, Carol would tell you. Critical thing is it's difficult to study just a monophenotype. It's almost impossible, and a lot significant subpopulation of our heart failure patients, they also have diabetes, metabolic disorders, and that's we also cannot separate. So I mean, with that. It's also part of the reason I think the FDA is interested in those studies.
Yeah, to tell you that I am the personal investigator of one of the big NHLBI cohort studies called the NASA study, which is 1/4 African American for Black, 1/4 white, and 1/4 Hispanic, and 1/4 Asian. We've done ancestral informative markers of all of our participants, and if you are self-identified as Black, your ancestral informative markers say that you're really Black. If you're self-identified as Hispanic, it's all over the place. Black, white, Asian. You're self-identified as Asian, fairly confident that you're ancestral informative markers say that you are. The Caucasians also all over the map.
We were talking about that with the Jackson Heart study recently, that self-reported means that we think you are. Yeah.
Right. All right. So since we're talking about clinical cohorts, let's move to you guys. I have a lot of experience working with these large, multi-center clinical cohorts, and there are unique issues in dealing with them. I wonder if either of you have had issues dealing with that in terms of blood-building or with Jackson Heart. So some of the issues that I've had is, we have to go through a central steering committee for everything, and that's difficult sometimes to get the kind of agreements we need, and also sometimes the funding agency has their own particulars about how you use their data.
Yeah, I know for the Jackson Heart study, like I was saying, for the ancillary study proposal, there is 4 layers of, I should use the word bureaucracy. And so, like, the first committee approves it, and then it goes to a sample committee that reviews it, and then another committee reviews it, and then the NHLBI reviews it. So you have like 4, levels of signatures before it can even go to IRB, and then IRB gets their hands on it. And then after that, you have to do a data PMDA. I still don't know what that means. You have to do that paperwork, and 4 layers have to sign off. So, like I said, and according to the website, 3 months it can be approved, not on Earth. So 3 months usually takes a year to.
The same thing we like as well.
usually requires an ancillary study.
Right.
With different lab committee institute.
Right. And we just started a workforce to try to streamline some of that. For example, if the IRB is expedited, can we truly have expedited RFP, or does that need to take 3 months also? So, you know, we're aware of those delays and trying to come up with ways to make it a little easier.
Are you aware of any unique issues with Wallerly?
For Wallerly, I guess the collecting data and the right person for it, and you can
Unfortunately, the SCSI colleagues
Yeah.
who were involved in knowing these patients weren't able to make it. So,
Okay. All right. One of the other things that I think all of us have thought about is when you're dealing with these large cohorts or even any clinical data, the people with sort of data science and data manipulation expertise aren't necessarily the same ones with clinical expertise. And trying to figure out how to get the two to speak is important. And have you encountered it? Again, you know, we've spoken about some of the issues with the work you're doing. Maybe you can.
I guess the question is more like, what about the data? Is the data information, but we should kind of distinguish for it, figure out which one is everything. Concentrate, try to link it physically, but one file is relevant. So I think the biggest challenge is actually from the master data, how can you inform the question, or how can you phrase the question that we're trying to do.
It's one of the beauties of this collaboration. Sitting in the meetings where you've presented has been fascinating. To me as a clinician, I thought, oh, we see this all the time. What does it mean?
Well, another thing when we chose means was that we don't know what they mean.
That's trying to mean something, so it's kind of nice the collaboration. What do you see?
Yeah, it's true. Whenever the clinicians come to the meetings and we go to their meetings and cross-talk, and it's always much better. It's when one side, one side comes in and just wants to look and see what the data show. If it's not question-driven or hypothesis-driven, then you're going to be limited in what you're able to do.
So I think these types of discussions are going to continue to be key and fruitful. I know that the fellows in the back who've done clinical work are really excited about a lot of the data science, and I know I am as well. Hopefully we can help each other come to some answers.
So as these studies get more complex and require multidisciplinary teams to come together to try and answer them, I'd love to hear the opinions of folks up there about how feasible it is for an individual lab or an individual research team to actually carry out a study from and actually bring together all of the expertise that's needed in a, whether it's a consortium or whether it's a university or an institution, or how I would suggest distributing those expertise.
Yeah. My thought is this type of consortium is absolutely what's going to be needed in the future. There's no way any one of us can answer all these questions alone, I don't think. But what do you guys think?
Anything other comments? So I have a quick comment as I also see the grant opportunities here. Clearly, we talked about at least 3 different set of cohorts, and we've seen how detailed and very elegant analysis of the elderly cohorts was by MyGene.info or MyVery.info analysis. These analysis really, if there's any way to learn these approaches, apply to the UCLA patient cohorts, or if we could provide that information to the Jackson cohorts, that's one big area that we can move things forward. The same thing about tools that we and have built, very powerful tools to discern phenotype differences and predicting patient outcomes. Any value of that to Jackson Heart study or to elderly study with either Andrew or Dr. Yates or Dr. Eric Koppel with some sort of brainstorming there, we learn from each other. I haven't had sufficient time to study all the information. You know, there's a lot.
available on the XMR page, but I have a feeling a lot of it's text-based. So how do we actually extract information from that and also adding omics analysis to that? I think if we could get some of those executed, we're doing our job to say the tools we've done that is validated and tested by more than one cohort. Because right now it's just one cohort. Most of these tools are fine.
So the text from the Jackson Hart cohort, those are what type of text? From physician record?
It's a combination. So Mike Griswold is the head of the coordinating center for Jackson Hart. So his group is the one who has all that data. So in terms of getting the identified data, we're approved for that. So we just need to work with his group to say, you know, if you want all 5,300, if you want, you know, and then whether the list of variables that they've accumulated, which of those you want. So, you know, it's just a matter of going through that process and letting them know what we want.
Mary, I think you alluded to the potential of generating proteomic or other omic data on this. Can you talk a little bit more about
Yeah,
that?
we put in the new grant that we're going to do proteomics on a subset. And so the cohort that we have planned to evaluate is to look at, in the Jackson Hart study, there's actually 99 subjects who at visit 2 had MI previous to visit 2, but did not have heart failure. And then by study 3 of those 99, 74 remained heart failure free, and 25 have progressed to heart failure. So getting those samples, plus having another 100 that have a cardiovascular disease that's not MI or not stroke, and 100 healthy controls. So that's what the comparison groups are going to be. Those 299 subjects, we're getting plasma from them, and then from visit 2, and then evaluating those. We're going to do a glycoproteomic screen, because then that will keep it, proteomics is still, you know, a little bit targeted.
Yeah, one thing that, you know, I don't know, I always have the desire, right, to have more omics to mention on single data sets, right? And some of the metabolomics work that presented, right, I think would be fantastic to see in that.
Yeah, and I've talked with Rob Birchwin on a separate project to do some metabolomic analysis of some of that cohort. I know he's approved to look at Framingham, and he's talked with Jim Wilson about also doing some metabolomics for Jackson. I don't, you know, we got to stay within our budget and our you know, what we've been assigned to do, but at the same point, a lot of that's coming out. Like, for example, the GWAS. You know, we'll have GWAS information on all of those subjects coming down the road.
Yes. So the database, the CARE database, is it the phenotypes following control vocabulary, or?
I'm sorry.
Are the phenotypes following control vocabulary, or is it more like a text description?
I am not the one to ask that.
I assume it's like the Mason study, there's a data dictionary, and they have a very standard, and NHLBI tends to use the same definitions for everything. There's probably a lot of we could do together with Mason.
Yeah, I know Mesa, Jackson Hart Study, Eric, they often get grouped together.
Yeah, they group together.
Yeah.
Great.
I also have a question for the clinician expert here. So, about like a patient's consent. When you guys get the consent from patient, after that, patient actually care about how the data say, involved in some study. Are they eager, say, like to get notified when this data set, this data get used as something like this?
It's a really good question. So in large NIH cohort studies, almost all of our consent forms say little checkboxes. They do want to know the results of this research. They do not want to know. They do want to, follow up. And whatever they check is whatever we do. So most of them say, yes, I want to find out whatever you find out from this research. So we send out periodic newsletters about what's going on, and 2 or 3 times a year.
I mean, we'll sit down individual and say, you have disease X.
We will send out
to their physician.
We do. We ask for checkboxes. Do you want the result of all of your lab studies?
check. We want us to send a copy to your doctor check. And if they have checked those, we will send back everything to them. The only thing that gets really tricky is with genomic data. I mean, if you have a genetic disease, and we are required to offer you a genetic counselor and give you all that stuff, if we tell you you have something again, to say, oh, bummer, we have to give them a lot of resources to deal with that, and that gets built into the grant and the money, and it's very expensive.
Yes, there's a lot of ethics to think about for this, when you prepare the IRB and the consent form.
Exactly. So for instance, with yours, if you happen to find metabolome that predicts death in the next 6 months, you're sort of obligated to then sort of inform, this is what we found. Not that we don't have to deal with it, but this is what we found. Great. Well, thank you guys very much for your discussion. Yeah. So it appears we ended a little bit early, which is not a bad thing. More time for informal discussions and big groups who want to talk. The coffee should be here at about 3, because that was a good break was planned. So just hang tight, you guys can hang out here or go up to the plaza. We are pursuing consortium training session until 3:30. Or would you like to finish that?
Could we push it up? We do have Penny and a couple of other people who need to be on a flight tonight. Right.
Well, the flight was cancelled, so you're
What?
We're going to take your time.
Actually, break 2 hours.
I'm so sorry. Is it because of storm in Europe?
No, they just said it was cancelled out of London, so it was
The plane was still in London when it should have already been on its way for 2 hours, and now it's canceled.
So I don't think we go and hope that it's another flight, or if we wait. I don't know. We haven't discussed it.
I checked. Well, let's.
Yeah.
That's the discussion.
So is everyone okay? We come back here at 3 instead of 3:30. We do a sort of 18-minute break and we come back because the other session, then we'll have more time. We have some very exciting and interesting talk in the consortium session on really new and fascinating things. So we come back a little early. That's okay. 3 o'clock?
Yep. Perfect. Thank you. If anybody would like some coffee, there is coffee on the table over there. And could everybody please take your seats relatively soon so we can start on our last session. Thank you.
So you're the next speaker? Okay.
Could everybody please take their seats so we can start the last session, please?
Okay, the first talk of the session will be Synapse by Brian Bott from Sage Bionetworks.
Thanks, everybody. So I'm gonna start by giving a little bit of background about Sage Bionetworks. This is oftentimes the first question I get when I talk to people: who is Sage Bionetworks? So we are a small nonprofit. We are in Seattle, Washington. We're housed in the Fred Hutchinson Cancer Research Center up there. There are about 40 of us in the organization. About half of the organization, we have about 20 people who do primary research. These tend to be computationally focused, so computational biologists, systems biologists, machine learning folks, physicists, and then we have a handful of MDs on staff as well. About a third of our organization is what we call our platform team, who's headed by Mike Kellen down here. He had to step out for a few minutes. He's at the meeting today. So he organizes this team of software engineers who are building software solutions to help in collaboration and fostering open and transparent research, which I'll talk about here in a little bit. The last six of the organization is leadership and support staff. So who are we? Sort of before we get to who we are right now, I think this first bit is important to understand why Sage was built. So it was started by Stephen Friend, who some of you may or may not know. But as an organization, we're focused on a world where biomedical research is going to fundamentally change to be more open and collaborative. That's sort of where we start from. We believe strongly enough in it that that is going to be the future, that is a given as far as we're concerned. And then we pilot a variety of components that are necessary to build what we like to think of as the scientific research commons, which is really this, what has been discussed a lot of today, this big data to knowledge ecosystem. And how do we get there? We support communities of researchers working collaboratively across disciplines, a few of which I'll touch on in this talk here. We offer a handful of software solutions. I'm only going to talk about one, which is most relevant to this audience, which is Synapse. And Synapse is built to support open scientific collaborations, as well as to aid in reproducible research. And it does that by enabling the sharing of resources, data, code, results, but most importantly, their relationships to one another. Provenance was brought up earlier today. That's sort of a benchmark in the work that we do. And Synapse really provides analysts. It's a platform that's built primarily for computational analysts, the people we like to talk about, who are comfortable in front of a blinking cursor. It provides them a mechanism to communicate what they're doing in real time, as opposed to just at the time of publication. So there are a lot of really good tools out there already. There are a number of big companies such as Amazon and Google who are providing web-scale solutions for things like data. There are places like GitHub who do a great job of providing a web interface to code versioning systems. And there are places like Figshare where you can share results. What we find that there is not a great space for is describing how all of those pieces relate to one another, other than in the narrative of a publication text.
This image over here is actually a provenance graph from Synapse. I'll show you a more detailed version here in a little bit. So Synapse for the novice is built on top of what we call open and RESTful API programmatic interfaces, such that the underlying data can be accessed through a number of clients. So we go to www.synapse.org. You can browse public projects that are out there. You can sign up for an account, build your own projects, which by default are actually private. You're the only person when you start a project who has access to what is within your project. But then you can share that with your sphere of collaborators, however narrow or broad they are. But importantly, it's not just a web portal. It's not just a website you go to. You can access these APIs through any programmatic client as well. So we have fully supported R, Python, and command line clients. I should note, a lot of people ask if Synapse computes for you. Synapse is not a compute store. Any compute that you do is either done locally or through your own resources. So essentially, Synapse is a registry where you are registering metadata about files that live somewhere. It versions those files, and then it allows you to point to the relationships, the provenance relationships about how something was generated. So at its core, it is a distributed and versioned file system where you can richly annotate the files that are there. So this is annotations on top of a file. So a file could be anything. It could be a PowerPoint presentation. It could be a CSV file. It could be a plot. And you're able to richly annotate, provide annotations on top of that. We also provide something called tables, which is an API accessible database, essentially, where you can have customizable schemas for structured data within a table, so within a tabular format. And ultimately, we're agnostic to a storage solution. So by default, if you quote-unquote store something in Synapse, on the back end, we are storing that on Amazon S3 and handling all the permissions around granting access to that. But there's no reason, we have in fact a number of use cases where people are pointing to local servers for really large files, to SFTP servers where they want to maintain another level of authentication on top of what we provide, as well as other cloud solutions such as Google Compute. And I think, importantly, on top of just having data and just having these annotations around it, we also provide a wiki authoring tool for people to sort of build up the narrative content that oftentimes goes along with these resources. And again, access controls so that you can share whatever you want with whomever you want. You can actually have portions of your project which are public and portions of your project which may be only accessible to a smaller number of collaborators. Governance, this was touched on earlier. I think ultimately this is going to be the differentiator between online platforms that make it within the biomedical space and those that don't. So it's great to see, for instance, EBI and their solution having a governance component to it as well. For Synapse, there is sort of a base level user registration and certification and user agreement. We also have a user certification process where in order for anybody to upload content into the system, you now have to pass a quiz. You have to get 9 out of 10 on this quiz correct. And the quiz essentially ensures that you understand as a user, one, some very, very basic sort of HIPAA privacy issues, as well as some intricacies about how Synapse works. So how permissions are handled, as well as how governance restrictions are applied to data. This was a tough thing for us to implement, I will tell you right now, because the software engineers and those of us who want to see sort of an open platform emerge from this are very resistant. We don't want to put any more barriers between a user and having access to the data. But at the same time, this is our hope is that this will eventually build an ecosystem of trusted participants who can sort of foster this community who will be responsible in handling these data. We have tiered access, so there is most of the data within Synapse is freely downloadable to anybody who comes. You actually don't need to go through this user certification to come and just consume data. You don't need to become user certified to contribute data. We provide a number of training modules and go through quarterly audits as well. Now I'm going to launch into a handful of examples of communities that are currently using the system and how it has become useful to them. So I'm sure most of you are familiar with the Cancer Genome Atlas, where they're exploring the spectrum of genomic changes involved in, I think it's close to 30 now, human cancers. And we were approached by their pan-cancer consortium about 2 years ago now, where they were trying to look at essentially molecular similarities and differences across the 12 most mature TCGA tumor types. So this is 6 different molecular profiling platforms that they were looking at. And the problem that they were running into is that, if any of you have access to TCGA data, awesome resource, but it's something that was essentially provided over an FTP server. The data was not versioned, it was not on scheduled releases, it essentially just rolled out over time. And so trying to amass, you'll see here in a second, a large number of researchers to all work on the same cohorts, the same subsets of patients, became very difficult. You could say, I downloaded this file from the website on October 10th, and then somebody on October 13th would go there and actually get something different. And so the Pan-Cancer Consortium, just as to provide some scope here, there were about 250 researchers who were on bi-weekly calls. I don't know how many people are on bi-weekly calls here. I don't know how many people can imagine having 250 people on a call. It was a little bit of a circus. Those 250 researchers were from about 30 institutions. There were 68 core projects that were proposed at the beginning of this. So essentially analyses and papers that would ultimately come out of this. And in the span of 10 months, these 68 core projects produced 18 Nature Publishing Group papers, leveraging the data that was centrally stored on Synapse. There were over 10,000 data sets that were distributed. To me, that's really not the important part. People can go a number of different places to get TCGA data, but what was interesting to see, and this is what we hoped we would see with these consortium, is that there were over 1,700 results that were shared back with the community during the course of this process. So it's essentially iterative steps that all of the groups didn't necessarily have to go through. I'll show you an example of that here in a second as well. So this is a paper that came out talking about how you bring together this many people to do this sort of large-scale collaboration. This guy, Cyriac Handoff, he was at Wash U. He was lead author on one of the 18 papers that came out. First author Nature paper, very impressive. But what we're starting to see from NCI and NIH is a different type of currency other than just papers. And so one of the things that Cyriac did, this is a screenshot of Synapse right here, is he took a number of the mutation analysis files, re-annotated them, took out duplicates, and made them all focus on the same reference genome. You can see here there were 4 different iterations of this that he did. And on the right here, you can see the provenance graph of exactly how he generated that. There's this algorithm called Music that he performed across these different types of files. You can see this is version 1 of this file, version 2 of this file. You can continue to crawl up this provenance tree and see exactly where this was derived from. And this specific Synapse ID, this DOI, in fact, was referenced in 6 of those 18 papers. So we're starting to see a currency also of data sets and curated data sets as works. So new NIH BioSketches has a new section called products, I think. So you can reference not just publications that you have published, but also software that has been reused by the community or data sets that you've made publicly available that are being used in other manners. Now, Syriac didn't create this data, but he did a whole heck of a lot of work to get it to a point where a number of groups found it to be useful. And so by providing this provenance stream, you can actually provide attribution all the way up to the data generator level. This is another example. The first one was really a group coming together around a common data set, common data that they were all interested in leveraging. This group all were trying to answer the same question. So there were actually 6 papers, these were 4 of them, that came out within the span of 7 months in 2013, all saying, we have the new colorectal subtyping, which is based on gene expression microarrays. Here it is, everybody. It's pretty cool. These are really, really good researchers. But what we saw coming out of this is that each team analyzed their own data set and came up with a subtype. And that first subtype that the first group came up with did not match the second one. There were different numbers of subtypes. One group had 3, one had 6, one had 4. All of them had different underlying prevalences. Each of them had different biological interpretations based on those subsets. And so it was unclear to the scientific community, to a colorectal cancer researcher or clinician, to understand really what to make of this. They didn't know if any of them were wrong, they didn't know if any of them were biased, or if it was really just different patient cohorts, or if it was due to different algorithms that were run. And so what we did is we reached out to these 6 groups and brought them together, and instead of each one of the groups running their algorithm on their own data set, we commonly curated them, normalized them within Synapse, as well as brought in publicly available data via Geo, and we had each team run their algorithms on every single one of those data sets, all 18 data sets that we pulled together. And now you can start to see subtypes still not agreeing across data sets, but as you have each algorithm, each expert team conducting their algorithm on all these data sets, you have this factorial design, and you can start to tease apart why these differences are there. Are they due to differences in samples? Are they due to differences in pre-processing methods? Are they due to different algorithms? Or are they just adjacent truths? This is a screenshot of part of the Synapse project. Again, you can drill into some of these files and you can see a provenance representation of how these were generated. And so this is my colleague Justin Ganay. You can see each of these little icons. These are actually GitHub icons. So there's code stored in GitHub. There are files stored within Synapse. You can keep crawling up the provenance tree. You can see I did a QC analysis as we combined the rectal and the colon samples within TCGA to make sure there weren't any batch effects. And you can go all the way up to Kyle Elrod, who's in Josh Stewart's lab down at UCSC, who works at TCGA, who did the initial import directly from the TCGA web portal. And you can drill into each one of these. You can see this is a specific commit of a specific file, a specific script that my colleague Justin ran. You can see the commit right here that he generated to produce those subtypes. You can start to really drill into what's underlying this. This is not something that Synapse records for you as a user. This is user provided. So this is something where Justin, as he uploaded that file, he said, I used these 3 pieces of code and these 2 pieces of data. It's important. It's an important distinction from like a workflow engine, something like Galaxy or Taverna, where it's much more modularized pieces of code that are meant to be rerun over and over and over again and really provide a processing pipeline. This is more a way for analysts to describe what they're doing and communicate it to either the people they're working with or the public at large. So the last example I'm going to hit on here, getting close to the end of time, is the Accelerating Medicines Partnership. So this is sort of is Francis Collins, director of the NIH. This is a really large public-private partnership between NIH, biopharma, and a number of nonprofits. To give you a scope, we're talking about, well, this was about a year ago. It was at $230 million. I think they've raised another 60 or 70 million from pharma. But really, the concept is to go after prevalent diseases, in this case, Alzheimer's, type 2 diabetes, and rheumatoid arthritis, where the underlying biological mechanisms aren't very well understood and where treatments are not always very effective. So pharma is wanting to understand better what targets they should go after. Academia is looking to do research. And what NIH wants to do is provide a public resource for each one of these types of data. We are interfacing with them on the Alzheimer's disease portion of this. There were 4 consortium who were funded between $10 and $15 million over 5 years to generate omics-type data on these post-mortem brain samples to both develop targets as well as validate them. So here are some of the institutions. There's one at Mount Sinai, there's one with Mayo Clinic, University of Florida, and ISB, one at the Broad and Rush, and one at Emory. Each one of these groups had their own research proposal. The FNIH came to us and they said, for this granting mechanism, we're going to require that these people, all of these institutions, make these data immediately publicly available. It was really, really interesting to be at the kickoff meeting for this when each of the grantees realized that when they had signed this, they saw $10 million, $15 million, and signed their name to it. It was a shock to some of them. I don't know that some of them realized that this was going to be the case. And so we were brought in sort of as a neutral arbiter between the biopharmas, between the academic institutions and FNIH to coordinate these groups and the data releases. And in fact, my phone's been buzzing all day because next Monday is the first data release. So come check out Synapse on Monday. There will be a ton of, I think the first set is mostly RNA-seq data that's going to be made available. And every 3 months there will be a data release from these groups. We're also coordinating an integrated network analysis for target discovery across these groups. So after they started to warm up to the idea that this data was going to be out there, I think the wheels started turning. It was like, okay, well, how can we best leverage this resource that we're going to have some, a little bit of lead time towards to really make an impact. And so now these 4 groups are actually dedicating analysts to do cross-project network analyses to aid in the target discovery. I'm going to skip through some of these slides a little quick because we're running out of time. But one important part about this is that each one of these granting mechanisms has a different way of validating targets. Some targets are more easily validated within certain model systems as others. So part of what we're going to be doing in conjunction with the FNIH and the biopharmas is to sort of be a broker at this stage where we're going to be sharing information about what targets are identified within each one of these 4 groups, and essentially doling those out to each one of the 4 groups based on what targets are most appropriate for their validation methods. We'll see how that goes. This is just a schematic of how the consortium is working. So there's a large consortium space within Synapse, which is private to the consortium. This is the collaborative workspace I was talking about where they're doing that cross-team network analysis. Then each individual partner has their own project they're working within as well. And then there are quarterly depositions out to this public space where ultimately results will be pumped as well, not just, again, underlying data. And this is where I was going to give a demo, but I'm on the docket for Friday's call, and so if you want a demo of the system, here's a plug. Tune in on Friday. Thanks.
So we'll hold questions until we get to the panel discussion. So faculty on hackathons, what, why, and how? Benjamin, good.
Hello, my name is Ben. I'm from Scripps Research. I work with Andrew Sue. I'm now an expert on hackathons since I ran one in November. Has anyone here ever been to a hackathon? So actually, more than I thought. So great. So for those of you who haven't, I'm here to explain what they are. And inform you that we'll be running approximately 2 of them a year as part of this instructions activities. And so we will figure out how to do this as we go forward. So here are pictures from 2 hackathons. This one is run by the Wikimedia Foundation. This one's by TechCrunch. Clearly they have more money to give to people to come, but both were productive events, so hopefully we can emulate them. And so the point of the hackathon for the participants is to build something, to meet people, and to have fun. In my experience, I had the opportunity to do a couple, they're called unconferences, where you basically show up, there's no agenda, when you arrive, you decide the agenda for the conference, and then you go and have your conference. So to me, a hackathon is very much like that experience, because it's very sort of loosely organized. In addition, you get to make something in the process, which makes it kind of more fun, actually. So from the, why are we doing this perspective at a little higher level, the hackathons are for a few things. Maybe most importantly, there's this concept of collaborative creativity. And so to give an example of that, it's often rolled out that apparently the Facebook like button was conceived and first implemented at a internal hackathon by Facebook. And there are many other examples in industry hackathons and things like that. Values that you do. Hopefully we'll come up with something better than that. The other thing to say is that it's a great opportunity to learn, especially a new API, and to advertise your API. So one of the things we could imagine doing is taking the Synapse API and running a hackathon around that. So what can you make with this? And it really gets it out there and gets people to use it. The other great thing about it is it's just a really interesting way, just like sort of like a different conference, like I said, of networking amongst really diverse groups of people. And so, the purpose for us, I assume why they want to do this, is that if you look, I just sold these, out of the participants in the Friday meetings. These are just a sampling of the tools that people have assembled and simply a large number of people underneath these tools that might contribute to a hackathon. And in addition to these people, we also have the sort of the greater community that we hope to serve and become a part of. And the question is, what can we do if we bring these things together, right? So can we make something completely new? Can we make individual services better through the efforts and the code that exists in all these subcomponents? So this is the idea. The big question is, what about it? I don't know. So Andrew already gave us talk this morning about the network file things concept. And this is really, so I don't need to explain it. This is a very, you can get this already, a very broad umbrella for a lot of different activities. And part of its purpose was to bring together out of the woodwork people that can solve individual portions of these problems, put them together. And as part of doing that, we have a mailing list, and we've got people talking, and we've run 2 hackathons so far. So I'm really just going to run through our experiences with these 2 hackathons, and hopefully opens up to some discussion and questions, especially for the people that have run or experienced hackathons. Eric actually ran one yesterday, so he can give his experience. So, first hackathon for network biobanks was at Stanford. It happened in May of last year. We had about 30 people show up from a variety of places, not only the West Coast, but notably Canada and St. Louis as well. We also had a number of random people that found the advertisement and showed up from all kinds of places, software engineers mostly, and students. To get people to come to hackathons, typically there are prizes. So in this case, we actually had $2,000 of prize money put up by Andrew's group and by Stanford. And there's four categories. One just for a use case, one for the most sustainable infrastructure design, and two for the best applications created at the hackathon. Because we had prizes, we had to determine how people were going to get them. In this case, we had a panel of experts judge the submissions and make their decisions. The experts being sort of the people like Arbuson at Stanford, people that put up money. In addition to the Stanford one, we tried to run one concurrently at Maastricht University in the Netherlands. This was a real challenge that we had this concept that we would have live video screens and chat and so forth, but it was really hard to do that. But we might be able to do that given some better technology in the future. So this is how it works. Hackathons, often you'll hear about them running for one day. They can run from a day to a week. Usually it's a couple days. In this case, it was a weekend event, so it started on Saturday morning, where we came in, gave introductions to the basic field and the concepts that we're after. And then, so the key thing about the hackathon to get it going is you have to have ideas for projects to work on. So people come and some of them will pitch an idea and say, I have this thing I want to make, please come help me make it. And those are a series of short presentations. Once people are done pitching their ideas, it's sort of loose, crazy chaos, and people run around and talk to people and figure out what team they want to be on. I think ideal team sizes are usually 2 to 3 people, although we had as many as 6 people on the team. The other aspect of hackathons sometimes are, as I mentioned, sort of learning opportunity. In this case, the NCPO gave a tutorial on their tools, which they're going to get people to use. And then we had lunch, and then people started hacking, basically started working to make what they had proposed to make. We broke for dinner and left after 7:30. Came back on Sunday. People worked more or less all day until 4:30. And at that time, you had to stop. You had to go and make a little presentation and sell your project so that hopefully you could win one of the prizes. And so that was the opportunity to rapidly assemble some slides, and if you had succeeded in making something that worked, give a demo, convince the judges that you were the best, and then award the prize. All of the code in this case was hosted at GitHub, which is an open source place for hosting code. And so you can actually still get to all of it if you're curious. And so out of that, those approximately 30 people, we pulled from them 5 projects. I'm not going to go into all of them. I'll just mention 2, one of which at the time was called the Network of Variant Annotation, which was led by Chun-Li Wu. And that turned into MyVariant.info, which you heard about today. The actual winner of the application, the best application at the meeting was something called Hypothesis, one of the 2 winners. And just to give you a feeling for the things that people worked on there at that meeting, the idea here was that, as it says there, given a hypothetical hypothesis, hypothetical drug disease association, propose a mechanism using known drug gene and disease gene interactions, and provide a visualization for that. And they actually put that together and had it running by the group, which was a really cool project. And so, but I'll say that everything, the MyVariant info was clearly the biggest win for this hackathon. So that didn't really exist at all before this got started. And through Chun-Li's ability to organize these people and get it done, it is now something that is a fundamental part of our lab's work and hopefully about to be a funded R1 by itself. And just to mention that, here's Tony from TSRI. The rest of his team is from St. Louis, Berkeley, Canada, Seattle, and San Diego. So this is what we want to achieve, things like this. And that was one of the reasons why we went ahead and did another one closer to home. And so this is the one that I was primarily responsible for organizing, so I know more about this one. So this happened in November. We had, again, it ended up being around 30 people, similar sort of West Coast focus, although again we had people coming from Washington University again and from UC up in Vancouver. And again, we had some pretty random people on the street, which was actually fantastic. One of the things I did as part of my duties promoting it was post on the San Diego iOS developers meetup group. And so we had a couple people come in from that, which turned out to be really great coders. And it was really a fun connection to make out of the scientific community to the industry. We again had prizes, which we used to draw people in. Less this time, but it seemed to be equally motivational. And in this case, since I don't like expert judging, I had the participants in the hackathon vote on the best projects at the end, and that's how we decided to do the prizes. And that worked fine. And so before the meeting, as I mentioned, I did some work on this. I spent quite a bit of time scanning our email list, sort of dragging people out of the woodwork, trying to get them to come, making flyers, having people post them on the university and so forth. One of the important things of a hackathon, as I mentioned, is that you have to have ideas to hack on. And so one of the things pre-meeting was to assemble this Google document with a list of potential project ideas which we see from our group, and we use to kind of explain the idea to people, and we hope that to get other people to add to. So as part of early work. This meeting went a little bit differently. So it started on Friday instead of Saturday, and it started at night with the primary social component here of this meeting. So we began, when people arrived, we handed them drinks and Indian food, and we let them talk to each other for an hour. Just that was the start. And then we went into the project pitch phase, and we let the rest of the evening be social. Talk to each other, form your teams, and make a plan for the next day. That's all we did that night. The next day, Saturday, we start at 8 o'clock, the doors are open, food is brought in, breakfast is brought in, lunch is brought in, dinner is brought in. They don't leave, they stay there and they work. They stay there until 10:30 in some cases, when we booted them out so we could go home. In other hackathons, typically ones running universities often they're 24 hours of like torture events where people will stay up through the whole night and do as much as they can. Well, we're too old for that. But this is a really productive day. And I think the setup on Friday and having everything brought to the people really kept them going and actually got something done. One of those real challenges is this is an incredibly short amount of time to get anything real accomplished. And so you want to be as focused as you can during that time. Sunday, 8 o'clock, doors open again, a couple hours really just to basically make sure their presentations, demos. They gave the talks, and then we voted, and gave out the prizes. We ended early because everyone had to go to another conference at 12, so that was that. The responses from all the participants were really positive about this particular one. Everyone had a good time, and everyone felt like they really achieved something. So we got more projects this time out of the same number of people. We had smaller project teams. I mentioned I think it's really the best way to go is trying to limit it down. I'm not going to go through all of these, but just to name a couple that were cool. So biopolymer was really driven by this guy, Mark Porter, who happened to have won one of the other prizes in the previous hackathon best use case. He's a local developer. He's not an academic, not funded by our grants, but he works on JavaScript and other web components for biology, and he came and put this project together with one of the Cytoscape developers. So that was cool. I was part of this information extraction project. Here's another example of a different kind of thing. So Civic is a big public resource that's about to be released by Washington University. It's already a big piece of software, and they came and they were hoping to basically just add bits and pieces to it to make it better. Citizen science is something that Andrew was a part of. We'll talk about that in a second. This one was completely different. This was from a student group at UCSD who were involved in synthetic biology, so completely outside of what we had been doing, but it's a really interesting component. NDEX is another big project run by a group at UCSD. I'll let Eric talk about the physio one if he wants to. And then we have another BD2K project. And so the winner here, now we have a prize too, which we'll use in the next hackathon, was Andrew's project actually, who worked in collaboration with my dad, who is a local developer. He's a software guy, and Hans Nieder for ECSF. And what they did, they actually had two different things that came out of this. One was an extension to a web-based text annotation tool that basically added what you see here, the ability to say, given a highlighted text, to classify anything that it was. And then my father actually created an iOS version of that, so that you could do that kind of thing on your iPad. And all that was working by the end of the meeting, which was great. The other one, this one did not actually win the prize. The student group from UCC with the synthetic biology won the other prize. But I wanted to point this out because this is, again, feedback from the participants. This was a huge win for their development efforts. So this is their website, which I can't show you the URL of because it's top secret, but it's about to come out. And this component of it was something that this is a gene summary, something that they were working on themselves. They had a database infrastructure set up to capture the gene annotations and keep them up to date. They're starting to work on that. And then we introduced them to the BD2K service, and they were able to basically chuck all of that code, replace it with calls to that API, and have it working by the end of the weekend, which saved them really a huge amount of time. So they were really excited about that. The other thing I mentioned was the iOS developers that came. One of them was the president of the company, and he happened to be also a Ruby developer, even though that's not why he came. And he ended up helping them with the problems in the back end of their website. So they were really pleased with the outcome of the weekend. And I would say for people that coming to future hackathons, it's important to come with a plan. And they came with a plan. They knew what they were after when they were coming, and they got it. So that was great. One of the wins I want to point out here, and this is particularly for the PIs and so forth in the group. So this is Dexter. He runs the Index project. He's a pretty senior guy, and he didn't succeed in recruiting anyone to his project. But he was very happy to basically be left alone, sit in the corner, and actually work on the code to make it work himself, rather than spending his time as a manager. So this was sort of a working vacation for him, and he was also very pleased about the end of it. I think Andrew also resonates with that idea. So wrapping up, this is the newly formed 6Gs of running a hackathon that I came up with. The most important one is this. You have to do a lot of the work in the beginning to get people to show up. And I would emphasize the need and the benefit of reaching out to your local community to get people that aren't normally in the room with you, because it comes up with all kinds of interesting new things that you wouldn't otherwise get, which is the main point, really. It's a social event, so you have to have ways of getting them to talk to each other. Drinks can help, but it's up to you to figure that out. Pick your project teams, get them working, and I should add, keep them working is important. One of the problems was that the first hackathon, I think, was that they took us out to what was actually a very nice, enjoyable dinner, but it really broke the flow of work right when people were just starting to figure things out. And so rather than doing that, bringing the food to them, keeping them locked up is the way to go if you want them to be productive. Get them to present their work and figure out some way to give them the prizes. At the end of the day, most people thought the prizes were a little bit silly. The students like the money. But it's really useful at this phase of the game when you're trying to get people to show up, especially when you're trying to market to undergraduates and people like that. So I think it is worth doing the prizes. So as I mentioned, just finishing up, we're going to be running 2 hackathons a year. The first one's coming up in a couple months at UCSD. Our group will be organizing that one again. Perhaps not me, but we'll see. These will be jointly sponsored by our consortium and CEDAR, which is run at Stanford, and the Data Discovery Index at CCC, all UCLA centers. There apparently is now some funding for travel awards to bring people to these meetings, which we really hope to use to increase the diversity of the attendees. So if you've ever been, those who've been to Hackathon or saw my pictures of it, they're kind of like the nerdy part of computer science departments. It would be great if it didn't look quite like that. If it looked more like the leadership of this portion, it would be great. I think that's all I have.
Okay, we'll wait for the end to ask questions. That's very interesting talk about.
We need to do hackathons at your APS meeting.
That's right. I'm thinking to do if there's a grant writing event. We could get that done one day right here. Or one weekend. Okay, the final presentation in this session will be the EBI Education and Training Portal. And today at lunch, Carol and I met with a bunch of folks who are interested in the training component, and we're all anxiously awaiting this talk. Because Kenneth, how do you say your last name?
How? It's a Norwegian name, so it's fine. It's perfect.
Is going to tell us about the resources that EBI has for training.
Well, as you can see from the group I'm working at DBI, I don't work for the training department as such. We'll try to understand what we do in the group training at DBI. First, with the numbers. I was able to get the updated numbers for 2014, but these are face-to-face. So these are people we have trained face-to-face in 2013, and as you can see, there's quite a large number of people, quite a large number of places around the world. So each group at EBI, each main service provider will have people within the team that goes out, that trains locally at EBI or goes out to different organizations and does training. I have updated numbers for 2014, so our online training portal had about, on average, 17,000 users per month. So that is what a decent number, well, so weekly. So training is very important because we try to teach people what type of data do you actually have and what can you do with this particular data. So our training is typically not to teach somebody how to become a bioinformatician, but it's to teach people that we believe may have some use for our services, our applications, or our data, so we can teach them then how to get the most out of the data we have. We currently have something like 60 petabytes of data on campus, and also this is within all the different omics, so not all of that is of course used in a training capacity. There are three main forms of training. The on-site is we have dedicated classrooms, so we have a dedicated training team, so they will then work with the domain experts and we give the different training. We have the off-site training, which is where somebody comes to us and says, can you come tell us what you do in this approach, or can you come to us for a couple of days and get interested in this field? So we then put together an agenda and travel out and train people. So when we do that, we don't charge for our time. So that's obviously the cost for travel is to come from somewhere. And then we have the online training portal, which I guess is more what this course is focused on. Just so we cover everything, the on-site training courses are very much hands-on. So we will sit people down with our data, with our tools, and it will be a lot of do this with the data, go on to the real research, do something, so try with data. We also have sometimes, which takes a bit more organization, but sometimes we say bring your own data, and we'll see if we can use that for training. Personally, I haven't had much experience with that. I know that other groups are doing this. As I said, this is a very, very hands-on and practical session. This is actually an example. This is a course we're in metabolomics all next week. And we put up a program rather than once, or a detailed program. People have to apply. In this course, it's a 10-to-1 oversubscription. So we have 30 places, we have 300 applicants, and select this applicant, interview them for this process to make sure that the people in the room have complementary skill sets, so that the training has a good flow. After the training has completed, all the training materials and tools and data used are publicly available. Then the offsite training, as I mentioned, this is where we are asked to go out. We typically have a concept called a roadshow. So we will go to a part of the world and we will have a week where we then give different types of training just to make sure that it's the most cost efficient and the best targeted training we can do. And that is the individual groups that work with the data, with the tools that go out and deliver the training. The online training, of course, is all done on the online portal, so if you don't at any given time. Each training course has the prerequisite, which is recommended prerequisite before the course, so that you kind of know that you will get the value out of it. And there's a few quizzes as you go along, just sit at the people actually trying to pay attention. The online training portal. This is just a screenshot, but what is interesting, as with everything else on DBI, it's all the data is completely open to be used for any purpose. So then, how do you find the training you're after? Internally in DBI, the different service teams are organized in these clusters. So that this is, when you're interested in a specific area, you don't need to go to a long list of all the things. You have to go into more of a focus area, and that will guide you further into the specifics you're after. So, just for people to get a feel for it, this is what you're actually after when you want to have some training. We have this quick course, which is literally just like a 15-20 minute introduction to something. And then when you go through that, and then you typically will progress onto more detailed ones. So in this case, they will typically around an hour. Or we have training that is specifically focused on more of a scientific area, or like areas it's conceptual about proteomics and metabolomics, etc. The video courses there has been training given. This is all being filmed and we put online, so you can go out anytime and see that. But we also have the webinars. That is when we announce that next week at 2 o'clock there will be 2 hours of something, and you can then go online and follow it there, or you can just go on with it later at your leisure. These quick tours, every resource we have are copied in those. But then we get into the more longer, more detailed courses, and it shouldn't be a surprise that genomics and proteomics are the areas where we have most training to offer on the online portal. But obviously this is growing all the time. It has been mentioned here earlier, so because of that there's more focus, for instance, on the PhD work. And they are putting together a chemical biology course. That's a simple search on the training online site, of course, with a passive search on the site, which we just enable to several different areas. So this is just what we would expect, of course. So that's nothing special about that. So here, for instance, we have how you submit the proteome exchange via Pride. This is what Henning touched upon. But then there's a small steps there. This will take about an hour and a half. So this is a typical introduction to something style course. And then we have one here for metabolomics. And again, the different stages, and you can obviously jump in and out at any given stage. You don't need to register to take a training course, but if the levels do, then you can subscribe to like a newsletter, and you can also then get a record of the courses you've done, and we also now are changing so there will be a bit more of a certificate you can get when you have finished a course. The interesting thing is there that we ask if you register, are you then willing to maybe do some usability testing, and we do get quite a lot of positive feedback, so that's quite good. So that is the link you asked about earlier, I guess. Yes, since I just asked to present a little bit of training, this is actually all I have in terms of slides. Of course, you have to talk about any questions.
Can I ask the other two speakers back on stage? So I think that's a short Q&A session now. So, yes.
I have a question on the hexagon. One is the next one scheduled with
It has not been yet.
It has not been
But
yet.
the goal is to do it before the summer, so sometime this spring.
So I know I've already seen interest in, from UCLA that we very much want to participate. There's students from computer science
we have additional information. Yeah, and you mentioned the idea of an LA area BD2K center meeting, since there are 3 in the LA area, and that would be another impactful thing to try to promote cross-fertilization of ideas.
I just actually judged a hackathon, which is called the Codathon, sponsored by the Hillary Clinton Foundation. It was all women. So we should talk.
I have a question about hackathon as well. I mean, there's something tangible that comes out, like especially if it's an R1 proposal, it's not great. I feel like I'm great people participated in listed individuals from different universities.
Well, I mean, let's be clear that the R1 proposal itself didn't.
But
it's
that's
an idea.
an example, of course, yeah. But if it's something that's useful.
I mean, so I think a lot of other people ask me similar questions. You know, why would I go to work for free? That's what they, the sort of developers ask. And you know, part of the thing that you buy into when you go to a hackathon, one of the needs is that you're going to produce open source code that can be used by other people. As far as, you know, if you're concerned about your ideas being stolen, then maybe you don't belong at a hackathon.
But you don't bring that idea to a hackathon. Right?
That'd be another way to say it, yeah.
And then, you know, any publication that results certainly will involve a partnership with whoever participates.
Yeah, and so I mean, maybe John could speak about this, but so in that specific case, you know, one of the other real drivers of this project was Sean Mooney, who is also going to be funded as part of that grant. And so that's a place to form the collaboration that could lead into shared monies and shared fame.
Just on that point, I actually think like Andrew and Ben created a GitHub repo where everybody commits, and I think that shows where that is. I think that's actually a much better way to demonstrate demonstrate when you had an idea and how far you developed it than maybe any other method. Because it's out there in the book. It's very clear, it's time stamped. Transmittal commit that. Right, attribution is right there. You know exactly which person is committed to what right?
So what do you do to define the scope of the book? So was it just a sort of one paragraph announcement, or was it much more detailed briefing? Because the list of topics was fairly broad.
It was very open. So, I mean, in the sort of material that went out beforehand and in the sort of brief introduction that Andrew gave at the first meeting, I gave at the second one, we sort of gave the broad scope. But again, the sort of the network of bioethics concept is so broad, it covers an enormous territory. And so we really wanted it to be a place where people could come and work underneath that umbrella slightly, but really just a place to go work together on something. So if someone showed up and what they wanted to do was come and eat our pizza but work on their private code, we didn't stop them from doing that. But basically people responded to the advertising around sort of the concepts that we're interested in, and it turned out that most people came and worked on it. It's if you look at industry hackathons that are more common, they will have much stricter guidelines about what they want because they're giving more money, they're pushing harder. So you will have to work within these guidelines in order to participate in the prize. So the other thing you could do is say, if you want the prize, do something within this box. If you wanted to show up and hang out, it's okay, do what you want.
Do you think bringing topics a bit together? Do you think a hackathon would be an interesting sort of final day or final 2 days to a more formal multi-day training course? Let's say we have a course at the EBI on networks and pathways, which is fairly formal, and it's one of the courses which Ken just described. So you have different pathway network resources, how we interact with them, and then I think it would be an interesting idea to say, okay, and once that is done, the formal part is done, then there's a 2-day hackathon. Do you think that people are too different and normally they would already be deep into the material, into the domain?
So I was actually just talking about this with Eric, and he was saying that it was a great thing to bring together training and this kind of productive thing. I would say that if you sort of require that, it changes the population there a little bit. So you're not going to have kind of developers who aren't at your training course beforehand showing up. But I think there's nothing wrong with that. I think it'd be a great objective for the course to get someone to the point where they could make something about. Or do an analysis. That's something I also should mention that other people, so the collaborators in Europe that have these bring your own data events. I don't know if you've heard of these entities, where maybe the point actually isn't to make code, but is actually to bring a couple data sets together that understand each other and start asking some questions and doing analysis work. I think that's similar or just a little bit different. Same kind of idea.
I've got one more question. I'm very self-interested, self-serving as well. So obviously this is at the time where the education data science and bioinformatics are of great interest to a lot of students on our campus. And as you said, Hackathon is offering training, EBI is offering training, and there's also information knowledge at Sage Networks. Posting the question to both Carol and Mary, is it possible we create a set of things and say if you accomplish 8 of these 20, you're going to get a certificate with data science? But how are people thinking in that line? So a lot of our students, they would be coming to get these trainings if at the end there is some sort of certificate.
What do you need? Is it just a certificate, just a piece of paper which basically any of us could print and sign? Or do you need really something which has credit value in terms of
So the certificate, I think, is the first step later on to get education credit, right? Or incorporate some of the things we do as part of regular curriculum. For example, at this moment, we're building a new master degree program at UCLA titled Medical Informatics. It's a master degree. It's not a PhD, and it's somewhat different. It's affiliated, different from bioinformatics. We haven't had any of the curriculum done yet. The words are out, and people are going crazy to apply for it already. And we are still in the process of figuring out what would be part of the curriculum. Education these days, everything is, there is reward, and because there is tuition for master degree. And how do we think will those contribute to the education as well as gathering resources for our research? And this is just, I'm sorry about this big topic for this discussion.
So I think Brian's talk actually really mentioned something very relevant to that, and the concept of having different ways of gaining credit. So he was talking about publishing papers versus publishing your data. I think that's something that could be taught to the students now, and so that one of the things that we look for in people that we're going to hire are contributions to open source code repositories, and so. You put that right from the center for undergraduates and said, well, if you want a job, it'd be useful to do this. That might be something useful. I don't know.
I totally agree. Just to expand on that a little bit, we have a few collaborators who are very interested in incorporating sort of a synapse and some of its functionality into course material, into graduate course material. And I think at a very basic level, it would be relatively easy to embed a lot of that into coursework, and then if they find use in it, then they can dive in and figure out a lot more of the more nuanced technical details.
When I talk to Sarah, I just mentioned that by email, they're training department in contact with European educational universities to see it.
As well as in the understand, BD2K director's office has contacted BI for training and as well.
There is in terms of accreditation of points, so our academic points.
So it's possible there is possibility of a certificate, or
there's hopefully
there are discussions about it.
Well, as far as I know, anybody who participates in these courses gets a certificate. That's, I think, already part of the course. So, but that's just paper, so that hasn't really any credit value. But there is, I think, a pending proposal, a grant proposal, I don't know if it's through or not, or building value, having credits for these courses in the context of the EU fairly transferable university credits. So that's not, it's definitely not yet operational. There's a plan for this, but it's not yet operational. But yes, there's also a discussion between BD2K and Elixir about a joint training portal, so something like potential here to do something jointly to make training and the efforts of the many activities from the different BD2K centers and EBI and eventually other Elixir partners accessible in one portal. But this is, I think, something which will take some work.
Just on a technical level, I think, I'm not sure if it's made it into the final grant or not, but we talked about doing, using something called the Badges protocol from the Mozilla Foundation. I don't know if you guys have done this, but the concept there is that it's sort of a semi-secure way of issuing a badge, like a certificate, through an online medium where it's under some level of control about who gets it, and so you can't just collect them without doing the work. And so that might be a flexible technical way to approach doing this kind of giving the credit in a way that's shareable. And that's something we talked about doing in the context of the educational games that we had proposed to create. So I don't know if that's you're nodding your head.
I haven't done
mentioned somewhere, I commented on one of the slides, DOI, so if we have design DOIs to make things citable, I assume.
So we allow users to mint them for objects that they think are broadly shareable. So we don't automatically mint one for every version of everything in the system. Honestly, our system is already set up using unique identifiers, synapse identifiers, but making it an official DOI and sprinkling metadata on top of it is important for broad sharing and resolving in perpetuity. And so yes, we allow users to mint those on a per version level.
And then the next step, are you already allowing or having infrastructure to allow users to assign their work with that DOI? Because that's now what's coming up, that you can, well, you can already, of course, the whole purpose is to assign papers with your DOI, but I think a big chance to make lots of this kind of works citable and assignable to your CV. The association of models is really a very good option. So are you doing that already?
We are, but it's on the roadmap, absolutely. Yeah.
That's really, I think that's also for crowdsourcing also even, I think that's really a nice option where you can allow people an easy way to say, hey, I've done this, I have done this small piece of work, because an orchid is only a number. It's really easy to say, okay, this is my contribution beyond just the usual publication list.
I have a follow-up question for Brian. So, I mean, a lot of Synapse is about creating these synergies between different groups, right? And I ideal world, right, if one group does that intermediate analysis and other people. So I imagine that collaboration process works sometimes, and it doesn't work a lot of the time too. Do you have any generalizable comments in terms of what characteristics happen?
Absolutely. And I tried to say it briefly, but I think the two main types of collaboration we found that work really well are people who are interested in coming together around a common set of data, usually a large corpus of data, where they might be able to share those intermediate results, but they may branch off into their own expertise. So that's one, something like the TCGA. And the other one is where people are highly, highly motivated about a specific question. And the colorectal subtyping consortium is another, is the other end of the spectrum, where these folks had their own data that they were generating, but they were all interested ultimately in answering the same question. And so those are two of the main types of groups where the collaboration can work well. And I will say too that it's not just Synapse isn't like making this happen, right? Like there's a lot of nurturing of these communities that go along with it. Synapse is sort of the tool that underlies those collaborations and allows them to operate as they did. So.
What's one that doesn't work well?
What's one that didn't work well?
In a generic term.
Yeah, absolutely. So there was something called the Federation that was brought together with a number of large, well-known PIs, including the CEO of our company, and they essentially had they thought if we just get the smartest postdocs, the smartest grad students together to answer some very generic question about biology, this is great. We'll get them to collaborate, they'll work together, but it wasn't very clearly defined on what data and or what question, and it fizzled. The incentives didn't align with those people either, so these were all postdocs who were nervous about, getting a first author paper and, ending up getting their career kicked off and working collaboratively across 8 groups. They didn't have the incentives didn't align for it to work. So that's an example of one that didn't work very well.
I have a question for Brian. So you probably mentioned that you have a slide, I just wanted to have a summary of that. If I have a data, say, maybe not necessarily my data, maybe some data available from Geo database or the RatePress, I say I want to work with other collaborator with this data set. How much involved to get this data set into the system?
As soon as you have an account and, like I mentioned, become a certified user, so those 10 questions, it's something you can post yourself.
It's like a completely done like a
much in the mode of GitHub, where as soon as you have a GitHub account and you want to start your own repo, you want to start your own project, go for it. And invite collaborators, invite those to come in. We want to work with you. And I will mention that it's very easy to point to data that lives at EDI or at Geo using a URL instead of redundantly storing it as well.
Also, is there an easy way, say, if I have a list of the geo dataset IP or re-express dataset IP, is there an easy way to tell how many are they already available? Like, is there a URL in Snap that I can link out?
So I think that the best way would be to search. Go for a search bar, and the search bar, I think right now it defaults to projects, but there's you can toggle it to all files or something like that. And if you go to all files and search for ESD 1234, it'll bubble up. Yeah.
So Synapse will store, if they NVM files, sort of value-added process files. Synapse stores those files in its own system, even with pretty large files?
Yes, it's whatever the user provides. It's agnostic what type of file it is, how large it is. I mean, you're limited by bandwidth, obviously. But yes, in general, with the collaborations that we support, we tend to not store very, very large files. But as soon as you get into the couple gigabyte range and down from there, that tends to be what the majority of people use Synapse for.
So what's the sort of sustainability, but also business model? So let's say somebody in TCGA did work. I guess your business model is you pay for usage of the platform, or what is actually the income? And then the next question is, okay, somebody did this, somebody generated some work which they want to refer to in the paper, they generated an EOI that refer to it from the paper, and then you store it in quote perpetuity for the money's flowing, or there has to be a continuous fee, or that's why you want all smaller files. So can you come with a bit on that side?
I'll say that's why we focus on small files. So as far as sustainability goes, we are nonprofits. We're not trying to make money, but we cover our costs. So up until this point, we have been funded by state grants, NIH grants such as this, and foundational grants as well. And by making the data open source, in theory, anybody else could take the Synapse stack and spin up their own instance as well. It's a public domain license. It's likely that in the future, so in the next couple of months, we're going to roll out the ability for you to allow Synapse to manage an S3 bucket that you own. And so essentially, we'll be deferring costs to the individual who's storing the data, and in fact, not taking any overhead on that. It's not like we're going to be charging 2 cents more. It's going to be something that they own, that each user is in charge of. So.
Okay. Any questions? Yes.
So we tried this collaboratory thing back in the late 90s. Hartwell was actually at a center that we were in. I think you may have heard Bob Franta was involved in creating the software to create this platform. The idea was that we had all these platform technologies at UW, proteomics, transcripts, all that stuff, and we would interact with various yeast biologists that were involved in a common problem like DNA damage, cell polarity, a few other things. And it was an interesting experiment in human interactions because the cell polarity group did incredibly well, worked together, contributed agents, shared data, blah, blah, blah. The idea was that with the data would be generated, we put in this collaboratory software tool, and then everybody that was in that particular consortium would have access to the data. The cell polarity one went great, headed by Dave Rubin. The DNA damage one was a complete disaster because there's too much at stake, I think. Because DNA damage and cancer and all this. And then lots of big names in this consortium, because we are, of course, would be wrong, absolutely. And people would send in samples that were just absolute garbage as a way to get into the consortium, just so they see what the competitors were doing. And it was
what? I heard a big name thing with the.
Well, I think it was an important area of science where huge careers are made. I mean, Lee Hartwell got a Nobel Prize for what he did in that area. So.
So as far as I'm concerned, it's all about finding the spaces where collaboration makes sense and where incentives align. Collaboration for the sake of collaboration is not
The technology never solves the human interaction problems. If people are already wanting, it just amplifies what's already there in terms of interpersonal dynamics. And if you don't have those fixed, there's not a software technology in the world that'll unfix it.
It's like the examples of what worked for collaboration and what didn't. It wasn't a technology thing that worked or a technology thing that didn't. It was really like how do these people, how are they going to interact with one another? They're all interested in the same underlying need, whether that be data or question.
And sometimes you just have to be really pragmatic and say what is the self-interest reason that's driving these people to work together? How do you engineer it such that it's really in their self-interest? Because the worst thing you can do is preach about how great collaboration is, because no one will ever come out and say, I'm against collaboration, I'm going to be selfish, and I'm not myself.
In the end, it's all about me. That's not a bad thing. It's just
that's how people get jobs.
It's how people have a career. Right? People are not going to sabotage your own career because you made a speech about the benefits of open data or open source or something, right? But if you're creative, you can sometimes find ways to align the incentives so that people get farther by open data.
there's some natural alliances that are made. Coders and factors. I mean, some things just make sense.
But do you think, those garbage samples that were submitted, that type of behavior have gone on in a completely open system? Like with GitHub, where it's totally open, and people start submitting these garbage pull requests.
Well, it was open to all the consortium.
Right, but completely open, or everyone.
Yeah, I think it's still.
You think they would still do it?
Yeah, but you see, the whole idea was that by contributing samples, you got access to the data.
What do you think? You could get access without contributing samples.
High-profile open source software project is going to have a peer review process, right? It's not just anyone can check it at any time, right? You have to be vetted by the community to become in that group of people that can add new developers to the project. If you're just initially involved in the project, you're submitting patches and then you're hoping that somebody is going to bring it all the way in. So, there have been some vetting samples and you weren't allowed in it yet. You need contributions right after. I don't know if the incentive to cheat them is so high, or at a certain point it becomes easier to actually contribute something of value.
The incentive to join the consortium was that you would get access to everybody's experiment in this particular area that would be relevant to your interests. I think if it was totally open, it probably wouldn't have worked either. I don't think it would have joined because there was no advantage.
That's essentially the Federation project that Brian mentioned, and that's another thing that kind of happened there was each of the PIs was in it because they used the other guys' data, so they were all trying to figure out what project can we do such that the other groups contribute their data and I don't have to contribute anything that I know that I can publish on myself, right? So the end result was nobody ended up contributing anything of value.
That's the other problem. You just join up just to get access to what somebody else knows.
Dr. Good, you touched on earlier about increasing diversity in computing science, and I think that the Axon started by the Foundation is definitely a good part of that. And I guess I just want to pose the question, all of you as a working with computing is perceived, and there's part of the demographics that will make up the base. Might be a big question, sorry, but I don't know, it's something I can think about in light of
I don't have a great answer to that.
Maybe I'll just cross this up. This was something that I was very, we saw this in hackathon number 1. I think they had 2 women, and I was like, oh, we can do better than this, right? And hackathon 2, there was a concerted effort. Okay, well, what can we do? And I've read a lot of blog posts on successful hackathons, and I would say that it's a hard problem, right? I mean, we have to just recognize that the field does have a gender divide. But I think the question is how can we be inviting into hackathon, be more inviting, right? So one for example, prizes I've heard, and I don't know the data to back this up, might be de-incentivizing women. Because it's more competition, right? It's like, oh, I don't want to go in, I don't want to be judged. I don't want to be espousing stereotypes here either, but the question is, is that a factor in whether or not hackathon is the person or not? Having them over a weekend, right? Is there differences there in terms of the participation? So, all I can say is we're trying different things, and we're very receptive to trying more things and hearing ideas from people impacting your situation. Right?
But that stereotype about men, they do it over weekend because they have no lives. Didn't you mention earlier on that there was consideration with these travel grants, and I sort of interpreted this that this was meant as an incentive to have more female participation. Was that the idea, to say they might be sort of widen the net by travel grants to?
I think that's the idea. I was just told of that yesterday. I don't know.
Well, one of the things that the Hillary Clinton Foundation did was they created a question that was more central to women. So they said, basically, how do you get people to live healthier lifestyles? So, and women tend to make their family health decisions and things like that. So the question that was central to women.
I think a lot of the data show that the source of the gender imbalance is middle school. Right. And if you look at pre-middle school, boys and girls are doing equally well in math and science. It's split in middle school, and by the time I'm trying to hire a developer and I'd like more women on my team, I've tried to get women in, and it's unreal. I can't change my hiring standards, and the applicant pool is extremely skewed towards men. It's really hard to do anything that far in. You got to go with the drop actually starts.
It's still a middle school problem for decades. And
I don't know what you do about it.
But efforts to try to fix that. I don't think
to address the now, trying to attract more women to these sorts of things, there are a couple of high-profile women sort of in this space. One is Kate Bainey. She's head of Mozilla Science, and she has a pretty broad network of engineers in general, but I think female engineers that follow her on Twitter, follow the work she's doing. And Jenna Polka is the other one who's at Code for America. And so, reaching out to some of these very high-profile people who might be able to get the word out to more females would be another way, short term, to attract more people to a hackathon, as opposed to trying to solve middle school education.
Right, and there's these scholarships, there's foundations, and Google, for instance, Google Coding for Women or something like that, where they give enormous scholarships for women that go to software engineering and computer science. You get solve the problem, as you said, an admission.
I don't think that this is easy. I don't think graduation rates in like CS departments, it's getting better than it was 20 years ago. It's not going to be 50%.
I can say something about
might be frustration, okay? For reasons that I don't generally, I think, not fine, okay? That's true. Also because, in fact, shooting is a very good career for ladies, okay? I kept telling them in daughters in the ladies. That's not because basically what happens is that, the guys end up, playing in front of the terminal all the time, they're sort of introverts, women go more extrovert. The result, they end up in middle-level management very quickly, okay? And I'm talking about middle-level management at the department school. I talk about Google, et cetera, where you get substantial salaries, you get, kindergarten for your kids, or unless you want to stay home and work from home in your kind of situation, you cannot even believe. So, but it's hard to change, and it's just changed slowly. I think the example role playing would be very important. In fact, I have good news there. The new dean of engineering school is going to be a woman. Okay? Yeah. Yeah. Okay, so we practice what we preach. Okay, at least we can say that much. On the other hand, we should learn for the medical field, but the medical field is to be exclusively male or predominantly male. But if I go to graduation ceremonies, there are a lot of probably more ladies than men. Is that correct or not? Yeah. And obviously, great results, okay?
There was like 15% women in my class, and there's now 50.5.
Which is wonderful, okay? So that's, what can we do? The time, but another thing is the more the computer go towards social media, the more attractive becomes probably, generally speaking, okay? Rather than just the very dry, the way it used to be in the past, right? So definitely you have to do the right thing if they are to.
I like the idea too of targeting the CEOs of companies that are women CEOs, like Xerox is headed by a woman. They do a lot of systems, stuff. So that might be a way to tap into the pipelines.
I also know like a successful story of a female developer. I went to a Silicon Valley, it's called like a developer out there, and it's a Python data conference. This is a society of called PyData. That's the society of Python developer, a female Python developer. They organize it very well. They like a regular meeting, have a certain activities, and they also do some like education for the high school student for the programming. So that type of activity, I guess, is also help the situation. I feel like it's also like social media propaganda in critical mass. Maybe for Axon, two women developers not enough. Maybe once we reach 5 or maybe then grow the word of mouth as we have more people. So I would say that there actually is a field of self-development, and it's public health. And so I was a student at the College of Public Health, and I would say it's about 80 to 20 kids. There are a lot of women in that field. And epidemiology is a field that we often, it's never talked about for big data, but they've been doing big data for a really, really long time. So I think that's a great field. UCLA has a public health school here where you can recruit some and most of them know SAS or so I think that's a resource that's underutilized.
Okay. Thank you. I need over some more questions to Dr. B for closing words or last agenda item.
I don't have any closing words, but I do think we have had a great 2 days of scientific interactions, vibrant discussions on different aspects of our projects related to the center milestones. I hope that everyone enjoyed it as much as I have. And Penny, could you say a few words about next year?
All I can say so far is that this meeting in quite exactly 1 year will take place in Cambridge. BDI will organize it, and it from the 22nd to the 23rd of February. And I hope there will be strong participation from all of you.
So what I could do from our perspective is there are some travel funds within the center budget. I'm going to work very hard to see if we could get additional travel support. If everyone wants to go, we figure out we will meet everyone at EPI. But before that, I know Ben and Andrew, you guys are going to have a hackathon coming up in the spring. There's also NIH meetings in November. So there's going to be a number of activities that Jennifer and Anders will post on the website. We could also start a female group if anyone's interested, or as well as a male group with email communications. It's wonderful to have everyone together in the same room for the past 2 days, and I'm just elated. I wonder, Carol and Mary, to offer something?
I just have had a fabulous time talking to all the people from different fields and perspectives, and I think it's going to be a fabulous collaboration. I'm very wonderful group.
Well, thank you all very much for coming. I certainly had a great time learning about all the different software studios and various efforts that we're taking around the world to have this big data center come to life. And I think that we're going to have a very successful year and several years to come, and definitely looking forward to the journey to Cambridge. So for this evening, you'll see that on your agenda, we've got a dinner at the Skylight Gardens at 6 p.m. We'd like for you to start heading that way and meet us around 5:30 in the bar area. So right as you come into the Skylight Gardens, it's to your left. And this is on, is that on Charter? I think that's on Hillbrook. It's just a few blocks away from your hotel, and it's, I suppose that I can.
It's on the Lindbergh. It's on the corner of Lindbergh and Lindbergh.
Is a rotunda, right?
Yeah. So if anyone is at Hillgard or the Government Hotel, I'll be in the lobby about 5:15, walking that way. That we could all walk there together, so we could have more discussions in that area. Now, in the BD2K Monday, we're really supposed to have 2 PI meetings a year. Year 1, we're only going to have 1 because we only have 7 months. So year 2, we definitely are going to Cambridge to have that one. And if there's any other ways, we could also have another small one.
Sure, maybe we can go have those with the NIH one.
Yeah, I was thinking that too. If we did November and then did the spring. Yeah, okay. And what about future years, Mary? Are you thinking if possible we go to Jackson? Yep, that would be great. Especially February. Currently, we haven't got a way to clearly identify everyone as part of the BD2K Center. We're going to look into possibilities of that and also look into possibilities of certificates and things in that nature for education purposes. We're going to organize education tours to different conferences. American Physiological Society and yours is conventional.
Oh, the American Society of Human Genetics.
Yes, and a number of other HAs, because we do have an education agenda in our center.
Yeah, talk about the workshop that we did. So end of March, early April, the FASTS meeting in Boston, and the American Physiological Society, the Saturday of the meeting, is having 2 workshops in the afternoon. One is on data, Judy Larkin and I are moderating it, and the second one's on proteomics. And so those are opportunities too, especially trainees. If you haven't been to FASTS, it's a great meeting
for career development in science.
So just for your information, we welcome your proposals. You could come in to say, hey, how about we host BD2K workshop at ACC next year, or any other society large conferences that you'd like to host a BD2K there, and we will be there for ASMS or wherever. So it's part of it. So we're charged with the mission to educate the broad scientific community.
Do you expect any BD2K 5 conference ever meeting?
I think they're getting their stuff together at NIH. I know they want to do like 11 center consortium. They talk about it consistently. Because we haven't seen the announcement. I've seen there's a number of NIH policies they're trying to sort it through, and I understand that. And my understanding is the finance, that part, we do at NIH, that's sorted out, that's solved. It's just policy issues to get things pushed. Everyone are aware of there are multiple RFAs on the BD2K website, right? And if you are applying for one of these additional grants, you need a center-wise letter of support or facility. Please do let us know. We will get all these. So see everyone.